Good morning, everybody, and a very warm welcome to dedicate the 75th birthday of Professor Emeritus Ülo Langel with the conference New Path to Pharmacology Using Cell-Penetrating Peptides. I'm very glad to welcome everybody here, and before I give the word to academician Anne Kahru, just a couple of words for the organizational part. So we will have now two parts of the conference. The coffee or light lunch break will be at 1, and we have a slight change in the program in the afternoon. So unfortunately, Professor Tambet Teesalu couldn't join, so the lunch break will be a little bit longer, 1 hour in total. So we will start after lunch break at 2 o'clock again. So the program then will be adjusted a little bit. But now I would like to give the floor to Professor Anne Kahru from the head of the Laboratory of Environmental Toxicology in National Institute of Chemical Physics and Biophysics, and she is the Secretary General of Estonian Academy of Sciences and accommodation here. So please, Anne, the floor is yours.
Respected speakers, guests, audience, austatud kuulajad, in my opening remarks, I will shortly stop on 3 moments. First, on our academy. Second, about this nice building, this palace. And finally, of course, the conference. Our Academy of Sciences is almost 90 years old. It was founded in 1938. It's a personal academy. It's an independent body of leading scientists and distinguished creative and performing artists who are elected based on their achievements. Currently, we have 77 members and 19 foreign members, including Professor Langel. We also have Estonian Young Academy of Sciences, established in 2017, and this body has 38 members currently. Now about this exceptionally nice building. It is a historic nobleman palace, and this person, that's very difficult to pronounce, Count Ewald Alexander Andreas von Ungern-Sternberg. He commissioned the construction of this building, and he is a descendant of a distinguished Baltic German family. However, he was born and also died in Estonia. He was born in Võnno Manor, Haapsalu, and surprise, surprise, he died in Elistvere. And this palace was designed by Berlin architect Martin Gropius. So currently this palace is nearly 160 years old, and we take good care of it, as you may observe. Now about the most important, this conference and the speakers. I did some homework. Of course, I went to Web of Science, and that is, I updated my knowledge. And as you know, this is an important database not only for scientists, but also, and even more, for science evaluators. So I had a good look, and I was really, I found that Professor Langel has close to 500 publications, close to 25,000 citations, and he has Hirsch index of 82. What impressive numbers, indeed. And I was very happy to learn that about half of his co-authors are from Estonia. And also, that is very important, more than 1,000 publications have been cited by patents. Today's program is very coherent with the scientific path of Professor Langel, and highlights the most important steps in his scientific career, includes the speakers who are scientific collaborators of Professor Langel, and also talking about the most interesting and innovative results. As biologist myself, I am very much looking forward for the presentations. I wish you a successful meeting. Soovin teile kõigile huvitavat konverentsi. Aitäh!
Thank you very much, Anne. So we will now start with the presentations, and the first presenter is Professor Tiit Land, who is the rector of Tallinn University of Technology. And Professor Land did the PhD in Stockholm University under the supervision of Professor Emeritus Tamas Partfai. And the title of today's talk is Ülo's Journey from Neuropeptides to Chimeric Peptides to Cell-Penetrating Peptides. So please, Tiit.
Thank you. So first of all, I would like to welcome you all to this conference, and I would like to thank all our guests traveling from abroad to present and to honor Ülo. I also would like to welcome Professor Tamas Partfai, who has been my PhD supervisor at Stockholm University, as Pirjo mentioned. So, and I chose the topic of my talk a little bit more maybe liberal, relaxed, about Ülo's journey from neuropeptides or peptides to chimeric peptides and to cell-penetrating peptides. And I have been working together with Ülo for quite some time. Ülo has been working on peptides almost for 40 years, and I believe with cell-penetrating peptides for almost 30 years. So Ülo has been my mentor, and also we're good friends. So I also will show you, present some maybe fun facts about Ülo and about our journey together, what we have been going through. But to start more, let's say formally, yes, as Anne already mentioned about Ülo's age index, but Ülo is graduated from the University of Tartu in chemistry. We both spent also quite some time in an old chemistry building at Tartu University. Today it's not anymore chemistry building. There is Faculty of Humanities today, and the building doesn't smell anymore of chemistry, organic chemicals. Yes, as Anne also mentioned, I also did my homework, and I actually checked this fact with Ülo, not to be embarrassed that he's the co-author of nearly 500 publications. I think nobody knows exactly what the number is because it's going more and more. I'm still going more and more. And yes, indeed, Ülo's age index is actually I found 88 in PubMed and 95 in Google Scholar, but well, anyway, nevertheless, this is okay. Yeah. Okay, yeah, but this is Ülo is in that terms is one of the definitely the most prominent scientists in Estonia. He has supervised 53 PhD theses. I don't know exactly how many in Sweden and how many in Estonia, but as Anne mentioned, Ülo has been not only active in Sweden. Well, we can consider him as Estonian scientist abroad, but in fact he has been very, very active also in Estonia at the University of Tartu, and he has also a number of PhD students supervised in Tartu. Yes, he's a co-author of 23 patents or patent applications. He's editor of 10 books, so all these are very, very impressive numbers. But now I'm starting Ülo's journey. So with this, I apologize for our guests who don't understand Estonian. This is new thesis in Estonian, and this is I found this. This is a print of a newspaper or journal or magazine of University of Tartu. in 1980. And you can just trust me that what is written here, that Ülo defended got his first doctoral degree in, this is in Estonian, structure and pH effects of cholinesterase in cholinesterase reactions. So yeah, effects of structure and pH in reactions of cholinesterase. So Ülo started his scientific work working with cholinesterases. And then he was, this was, yeah, as I say, before peptides. And as I said, we are good friends, and Ülo is always looking for something new. I don't know if he got bored of cholinesterases. Professor Jaak Järve was doing active work, good work still at those times at the University of Tartu on cholinesterases. There were also a number of PhD studies which were defended on this, but Ülo decided to move further, look at on newer avenues, what can be discovered. And then he moved to peptides, and he had the chance to, from Soviet Estonia, Tartu, move to Sweden. 1987 was the first time Ülo was able to travel to Sweden. I don't know how, I will tell my story, how I managed to go to Sweden. I don't know what was your story exactly? Where did you get your first visa, and what was your travel journey? From Tallinn to Helsinki by boat, from Helsinki to Stockholm by boat, because there were no airplanes. But that was 1987. Ülo took up a position at the University of Stockholm University, Professor Tamas Partwai's lab, which was already investigating neuropeptides and newly discovered neuropeptides. And also, what was very important, that Tamas' lab had skills, and also the machine. I will tell about machine also later, when I come to peptides, which are called M. Okay, I can tell you the story right now. If you, the peptides, what I will show, and also Tamas will show, Ülo will show, they are the many peptides which are called M. M15, M35, M40, Margus has M300, whatever, something. And if you wonder what this M means, they were obviously peptides which were synthesized chemically by solid phase peptide synthesis, but M means machine. That was Tamas' machine. Tamas had acquired from his friends, and the machine was in the lab of Stockholm University, Department of Biochemistry at that time. And those peptides, which are referred as M's, were synthesized by machine, but not manually. But Ülo synthesized, and myself also, we synthesized also quite few, or I would say many peptides manually as well. So yeah, that was really important that at that time there was solid phase peptide synthesis technique. We know that Bruce Merrifield got the Nobel Prize in 1982, perhaps, for this method he discovered. And yeah, and this is Tamas here some 30 years ago. I apologize, this is kind of party, so this is that we have some bottle of nice wine here, but we also had some fun at the lab, so we were not only working. So, and then first peptide was galanin, and what Ülo started to work with. And galanin was discovered by another prominent Estonian scientist abroad. Yeah, I said Ülo is not only Estonian scientist abroad, he's Estonian scientist both abroad and also in Estonia. But Professor Viktor Mutt was at Karolinska Institute, Estonian, who moved to Sweden with his parents after Second World War, and he actually discovered more than 50 biologically active peptides. And here is the photo which is taken right here in this hall 2 years ago in January 19, 2024, when we had similar conference to honor 100 years anniversary of Professor Viktor Mutt. And you can see here Ülo, as you can see Tamas here, then Tarmo Soomere was the president of Academy of Sciences, and Professor Mart Saarma, who is currently president of Academy of Sciences, Professor Toomas Högfeldt, Professor Jaak Järv, and nephew of Viktor Mutt, well-known writer in Estonia, Mihkelmutt, yeah. And then there's other people in the background as well. So, then I promised to tell you, and then in 1989, I graduated from the University of Tartu, also in chemistry, and I had a chance to go to Sweden, and still, the time was 1989, Soviet Union, and I also here, this is my first business trip, because this is служебный passport. Служебный in Russian means business. So I had a business passport, like probably Ülo and other Estonians as well who traveled to. Okay, you had red-covered passport. Okay, I had blue-covered passport. But anyway, so I managed to, and you can see there is in Russian in Kirillitsa. This is my father's name, but somehow I was let over the border in Sweden with this passport, and this was my first visa to Sweden. For 30 days, and I got it in Leningrad. That was St. Petersburg today. So I had to travel. I took a night bus from Tartu to Leningrad. I was next morning in Leningrad, went to Swedish consulate, got my first visa, and left Estonia for 30 days for Sweden, but then eventually I stayed 16 years, so but not only in Sweden, and then later in the United States, and also then back in Sweden. And then I moved back to Estonia when we didn't need any more visas because Estonia became member of European Union in 2004. I moved back to Estonia in 2006, 2007, so then the borders were open, but until that time, actually, we used to go every year at least once to police department in Stockholm to get visa or study visa, work permit renewed. But this was kind of our journey, and then we survived, we managed. So, and now our first studies, when I joined the lab, Ülo was providing us peptides. He was he synthesized peptides, both manually and then with machine. He had always, I don't have any kind of picture of Ülo with the lab coat, but I know Tamas has in his presentation. But Ülo was going in the lab around with a white lab coat. It had kind of touch of smell of organic chemicals, notably hydrogen fluoride, which was used to cleave off, used to cleave off protecting groups in the T-pop chemistry, and also from the resin. And that was our story. And basically, Ülo synthesized peptides, new peptides, and came up with new ideas what to synthesize. And I went to slaughterhouse, how to say. So we used those times rats, rat hypothalamus, hippocampus, but and also cell lines, and did ligand binding studies. So we also labeled these peptides by ourselves with radioactive iodine, which is not the case anymore today. But yeah, this was our case, and here is. This is the displacement of galanin and galanin fragments in rat hypothalamic membranes. Yes, we labeled this was tyrosine labeled with iodine, and so the galanin itself is here. Galanin 129, so 29 amino acid long peptide. The IC50 value, or this affinity value, was below 1 nanomolar. It had very high affinity, and in this picture we show that it's N-terminus of galanin, which is important for ligand binding, and also we had different studies also for biological activity. And galanin in that sense is kind of unique. Many other peptides have C-terminus, important for ligand binding. And this is 1 to 29, the full-length galanin. This is 1 to 16, which is about 10 times lower affinity towards galanin receptors. And this is galanin 17 to 29, so it doesn't have any affinity. And then when you mix together 1 to 16 and 17 to 29, though, there was no difference. the affinity was as for galanin 1 to 16. And then we carried also we synthesized the shorter galanin fragments, and galanin 1 to 13 is the shortest fragment which has a reasonable binding affinity for galanin receptors. So, and then next thing we did, we decided, knowing that 1 to 16 on N-terminal part of galanin is important for ligand binding, we made alanine scan of galanin 1 to 16 to identify important pharmacophores, amino acids, which are important for galanin binding. So once again, this is now, I am sitting here in the lab in the Department of Biochemistry, doing some calculations or preparing for an experiment. Ulo was constantly working, synthesizing new peptides, and just gave me peptides, and no, not only me actually, but we were to our group. And then we performed alanine scan. Once again, galanin 1 to the full-length galanin with KD below 1 nanomolar, galanin 1 to 16, and then we identified the most important pharmacophore as tryptophan 2. So when you replace tryptophan 2 with alanine, so basically there was a complete loss of binding affinity. And there are other pharmacophores as well. This arginine 5, tyrosine 9, and so on. So and then knowing that we could go further, then of course we discussed what to do next. And first what we did next, we went to kind of business trip again, because we also had fun. So this is in 1991 summer. We had done some good work. We decided to take a trip to Italy. Here you can see this is the car we were traveling. Lada, I don't know, some of you know, it's Lada 02. Yeah, and we were traveling 5 of us in this car. Ulo, his wife Ülle, and son Tõnu were here, and Tõnu has grown up actually, and my wife Kadri, and me myself. And when we visited Professor Peep Palumaa in Switzerland, who was doing postdoc in Zurich, then his basically landlord saw that the car was approaching, and then just more and more people coming out, and they asked him, how come? How can you all manage in this small car? But well, obviously they didn't perhaps know that we were from Soviet Union, and we could do everything. So, but trip was a lot of fun, but also business trip, because we visited in Milan our co-worker. I can't remember her name at the moment. Silvana, yeah, and who was doing at Mario Negri Institute. Exactly, yeah, thank you. And and then that was, as I told you, that was 1991 August, and when we returned from the trip, we drove all the way from Sweden down to Rome, and then back to Stockholm. And then when we returned, then a few days later, there was a change of regime in Soviet Union. Soviet Union collapsed. Estonia got rid of Soviet occupation, and then that was also something which gave us more enthusiasm and optimism to work further. So, and then now I'm coming to chimeric peptides. So the first chimeric peptide was M15, and once, yeah, this is I already told you what this M means. M means machine. And how did we come to this peptide or fusing together? That wasn't but something completely new. There were chimeric peptides had been synthesized also earlier, but as I mentioned, that the rationale was to take another neuropeptide, and as I mentioned, galanin has its as I showed, its N-terminus is important for ligand binding, and then we first peptide we came to substance P, which has C-terminus important for ligand binding. And so we fused together galanin 1 to 12, then or 1 to 13, because 13 amino acid is proline, and also in substance P, actually 5th amino acid is proline. So this can be galanin 1 to 12 proline, substance B4015 or whatever, but anyway, so, and we fused it together. And then when you did this is now in RIN-M5F cells, which is a pancreatic cell line, and we did a ligand binding study, then we were surprised that this is galanin 1-29, and this is M15, which was called galantide also at that time. There was a quite significant increase in ligand binding, and as you can see, this curve is not Hill coefficient, that this curve is not 1. It's bigger than 1. So there was also, we didn't at that time know exactly what to do with this, but anyway, this was, galanin M15, galanin, and galanin 1-13, and then galanin 1-13 plus substance B4011, so there's no difference. Substance B4011 doesn't bind to galanin receptors. And then also our collaborators showed that this peptide M15 actually was an antagonist. It reversed galanin-mediated inhibition of insulin release in pancreatic beta cells. So that was the first peptide. And then, I will not go through this through because my time is over and I will finish soon. But this also, I made a hypothesis or model how it binds. And then, this was 1991, and then we went on, and then in 1994, my work was done. For that time, I defended my PhD thesis. I don't know why I showed at that time we could show the color, which is your thesis is for the thesis. I don't know why I chose yellow. Maybe that it was dark time in January. I started to write up the thesis and I defended in May. But I defended my thesis and then moved on to United States because Tamas had convinced me that, or said me that you have to go to NIH to do a postdoc. I said, yes, of course. So I did, left the lab for first for 2 years, but I stayed almost 5 years. And then when I came back to Stockholm, then there was already huge, new story about cell-penetrating peptides. So I will not go into these because you will hear now more about, a lot about CPPs, but this is just one work where also I found myself being a co-author that was published in 2001, and as you all know, Transportan was the first cell-penetrating peptide, another chimeric peptide with mastoparan, galanin and mastoparan, ylosynthesized, and then the story went on and on and on. And then finally, I think one of the greatest honors to Ülo for doing his work is, this was 2 years ago, his French colleagues dedicated their book on cell-penetrating peptides to Ülo. So, so this was short story about Ülo story and our story together from early 80s to today. So thank you.
Thank you. Do we have any questions? We could have time for a quick question. Yeah.
Thank you very much for the nice talk. I have a question related to the chimeric peptide. Why did you choose substance P?
That's a very good question because the next neuropeptide was bradykinin, if I recall correct, M35. I think we chose substance P because Anders Sundén in Tamas lab actually worked with substance P. Maybe you have better memories.
Very small word at that time. Do you hear me? And I think the most remarkable thing in this solid phase peptide synthesis, you see the number of peptides he has synthesized and based his work on. Substance P, which is only 11 amino acids long, and was part of the Nobel Prize of Ulf von Euler, who got the prize for noradrenaline and substance P and prostaglandin. A pretty good price, I should say. Nevertheless, the composition, but not the sequence of substance P for 30 years was not solved. And substance P analogues were very hard to synthesize in the solution. And so when we got this machine, I will talk a little bit about it. It was made by Lee Hood, who made the first sequencing machines, and which really changed it. Today's AI people, they are eating data. And the data is the type of data which he has. If you make 50 alanine analogues changing, then so and so affinities will change in this or that tissue, meaning either that the receptor is different or that this pharmacophore is different. The very first mapping of a pharmacophore was made by Tiet, and I met Ulf von Euler, who was kind of the king of the Karolinska pharmacology at that time. And he said to me that I hear that Victor Muth is not making so much dirty smelling peptide extractions. And it is because he only does it once from a ton of intestine or a few hundred kilo of thalamus. After that, he goes to your place and you synthesize for him so much as it took for tons. I said, not only do we synthesize, but Udo Lange took it over, organized it, labels it, and he sets up the projects. And the two peptides they worked on was neuropeptide Y and galanin, which for the first time systematically the pharmacophores of a peptide were determined. This is not standard. So all the molecules you see, which are now for migraine and so on, they all come from this table of Tietz and Ülow of finding on a long peptide, which are the pharmacophores. And substance P, Ulf von Euler said, if you are to take another peptide, there is no question that it has to be substance P. And in those times, you didn't tell in the middle of the Karolinska Institute garden to Ulf von Euler, you are wrong. There are many others which are more interesting. You got your Nobel Prize for it 30 years ago. You didn't do that. I said, we will look at it, which part of substance P we use. He said, let me give you a slight suggestion. Don't use the N-terminus.
Thank you.
Thank you very much. So I think we will move on now. And now we are very honored to have the presentation by Ulo Langel, Professor Emeritus of the University of Stockholm and University of Tartu, Adjunct Professor of the Scripps Institute, Loyola, USA, Member of Academia Europaea and a Foreign Member of Estonian Academy of Sciences. So, Professor Langel, the floor is yours.
Thank you. Do you hear? Yes? First of all, I would like to thank the organizers. The idea came from Jaak Järv and Pirjo and Tallinn Technical University and Biochemical Society organized all this, and I maybe forgot someone. Many people did it. And I think we have here a very nice collection of speakers. Unfortunately, Alain Prochain couldn't come, but he asked me to forward his best regards and told me that he's better now. And thank you for giving me this opportunity to speak about myself. This is a privilege of older people, I think, That's why I got this privilege. I am talking, actually dreaming about the new pharmacology. Do I have this? What slide do I have? Title, yeah. I'm talking about new pharmacology, but actually this is a way to new pharmacology, we hope, that will use cell-penetrating peptides, as they are called today, and which are passing by barriers and can carry different drugs to different organs and tissues today. And when we started this in mid-90s, that was still a dream, and nobody believed that the peptides could penetrate cell membrane. And it was a nightmare to publish a paper on this, even if Alain Perchian already had published his penetrating peptide 1994. And I will remind you or tell you about the way or beginning even, and that's why I bring this up, because I want to tell you what I have learned on the way from the beginning. And also, you have a lot of names on these slides, and at the beginning it was a lot of names from Estonia, and then it started at Tartu University. And you can see the name Viktor Fedoseev, who was my first contact with the lab number 414, where all the most important biochemists of Estonia came. You see some names here. And Viktor Fedoseev was a guy who was playing basketball at Tartu University together with chemistry students, and I played basketball with him. And he came to me after a play once and said, "Listen, you are a good basketball player. Why don't you come to the lab and try some biochemistry also?" And I did. And it only continues. And you can see here nice names from the lab. We had a small corner of 414 only, but it was full of people all the time, and it was working day and night. And it was actually a bioorganic chemistry direction at Tartu University, which was initiated, I think, by Viktor Palm and Tullio Ilomets, who were enthusiasts in the field. Yeah, I'm correct? That it was Viktor and Tullio. And after that, I worked for a while with muscarinic receptors, which Jaak brought for Tamas lab to Tartu. And in '87, I moved to Stockholm. Again, you can see a lot of names. And I did a collaboration with Tartu University all the time, and was 1 year in Tamas lab in La Jolla, a Scripps Institute. Again, fantastic names. And in 2025, I was engaged at Taltech, in a board of Taltech, actually. And I find this also very interesting. So what did I learn during this journey? First of all, the first lab, I learned scientific enthusiasm. And if you don't have this enthusiasm, you will not be interested to continue, I believe. But I got this injection and I continued. When I studied for my first PhD, I studied cholinesterase by methods of chemical kinetics. And I bring here the example of phosphoorganic compounds. And this was Jaak, I think, who synthesized all of them. And some of them came from Moscow also. They had these preparations for Novichok, I guess, in Moscow. But on the way to that, we could do some science. And Jaak was this guy who smelled terribly when he came to 414 room and had made a synthesis. But he brought these compounds up to measure. And it was a period we didn't have much money. Actually, we didn't have any money. But we had time. And kinetics is about changing your time. You are measuring something in different time points. And time was what we had. So we measured this constants, and on this, we have this. Where do I show? Ah, here. Do I show? Do you see what I point? This. In this figure, the hydrophobicity is plotted from binding constant of these phosphoorganic compounds to the enzyme. And what you can see, there is a break in this graph. And that's what we interpreted as a determination of the size of a hydrophobic binding site. And we had all these characteristics, and we sometimes said something about the mechanism of interaction. But it was quite a few things which we could say with this method, because the system was very complicated, enzyme. And in this color figure, you can see that this anionic binding site is now determined by other methods for this enzyme, and it contains pi electron systems conjugated somehow in the enzyme structure. But in these years, when I was working with kinetics, we all guessed that this must be some aspartate or glutamate in active site. So what did I learn from that? You are always wrong until somebody, or you are always correct until somebody shows that you are wrong. And that even now holds. Yes. You agree? And this happens all the time, that the new things come up and then you have to change. But at this stage, it was a very good thing for us. And this was something we could do. Then, 87, I turned up in Tamas lab. And again, you have a lot of interesting names here, fantastic researchers who gave a lot of knowledge to everybody and to me as well. And it was a galanin binding and synthesis, as Tiit showed you already. The neuropeptide world was quite high up, and Sweden was in front of this in the world because of Victor Mutt and others. And I found one thing here in galanin field, which was also very interesting, that if you have a peptide ligand, neuropeptide, something, you are not good researcher if you don't have a non-peptide ligand to this receptor. And that's why we try to make different kinds of non-peptide, use non-peptide approaches. And here is one result of this. It's a gallinon, which I made based on a small combinatorial library. And it also gained a lot of attention from a scientific world. Non-peptide ligands for peptide receptors. And that is what the pharmacology is always interested in. When I come to chimeric peptides, I was using this M letter referring to a machine. I didn't realize for a while that manual had the same letter at the beginning, and so it is. You forgot to mention NPY or M32 analog, which has been studied less, but the characteristics of all of these chimeric peptides constructed from two neuropeptide sequences had extremely high affinity. They were below 1 nanomolar affinity very often. And we don't know exactly why. The explanation was this double binding to two receptors simultaneously. But actually, this is unsolved, this mechanism still. But it's a kind of synergism which you get when you connect these two sequences to each other. When I come to cell-penetrating peptides, which for me is based on this initial work with neuropeptide receptor ligands, and I bring here some names from Estonia on this slide, and some of these people are here today. And there's a huge number of Estonian scientists who are involved today in this cell-penetrating peptide research, at least on one end, if not more. And I think there are 20 students or 22 students who defended in Estonia in different CPP and Galanin works. And more of them, like 6 Estonian students, defended in Stockholm also. So it's approximately half of my PhD students who are connected to Estonian universities. And here is this figure again, how these chimeric peptides looked like, 2 different parts, Galanin on the left and something else on the right. And when I was trying to continue with this, to understand the Galanin receptor binding, I figured out that if I put something like mastoperon to the C-terminus instead of substance P or bradykinin, then I obtain something which penetrates the cell membrane. Mastoperon is a wasp venom toxin, and it was known to interact with the plasma membrane. It was toxic, but if I connected it to a Galanin part, then it started to penetrate the membrane somehow. And if you connect if you connect nothing. Okay, if you see this lysine, which is labeled with a star, if you connect something to this, what happened now? It started to live its own life. If we connect something in the middle of a molecule at this lysine side chain, then this transporter molecule was able to move this molecule into the cell as well. So it was an efficient cargo transport system which we discovered. I think it was Margus Pooga who made the first experiments with transport and delivery in Tartu, I guess, or Tallinn. It was in Stockholm, maybe. Okay. Well, we didn't. It was not easy to find a confocal microscope that time, so that's why I don't remember. But it was at least Margus I remember from this period. And first we made a longer transport, and since we formed this molecule from galanin, when we thought for a while that first N-terminal amino acids were holy and we cannot touch them. But when Ursel Soomets came to my lab and I asked him to make it shorter because it was too difficult to synthesize, and he just took away first 6 amino acids, and then we got transportan 10, which was as efficient as transportan was. And then he made a lot of work which Tiit showed before for galanin. He made the same structure-activity relationships for transportan. Huge work for synthesis. And this later led to our PEPFEX and NICFEX, where we have stereolated N-terminals. And I will show you what this is good for, PEPFEX and NICFEX. And we don't know actually how they work. We hear from Sandrine today some new mechanisms. But I think it's a mixture of different proteins which endocytose the peptide and peptide cargo. But it's also a transportation mechanism which is not followed by this. It's a direct translocation. And I think almost every bioactive molecule today has been transported into the cells and through barriers also often with these peptides, and some of them are listed here. Really huge antibody molecules, for example, and a lot of oligonucleotides are in this list. So everything which you can take in, you can use for influence of intracellular processes. And we start from connecting transportan on side chain with a peptide nucleic acid. This is also Margus who was first go over on this paper. And peptide nucleic acids was a very, very hot thing at the end of the 90s. And I got this idea from Asko Uri, who is here today also, who recommended me to work on this. And then we did, and we got a very good papers out of this. And it was a covalent connection of a peptide nucleic acid to a CPP side chain. And it was thought that if you get this into the cell, the disulfide bridge will be broken, and the free peptide nucleic acid will do its work. At this case, it was an antisense DNA sequence which we used, which recognized the targeted mRNA and made the translational arrest and arrest of protein synthesis. So you could stop or silence the protein synthesis in this way by sending in the peptide nucleic acid. And after that, it was clear that peptide nucleic acid is not the best one in the world. There are several other techniques, but siRNA was not discovered yet, and other techniques were on the way also, but were unknown this time. And here are the first pictures. You can see this is in a lower figures you can see the antisense effect of a calanin type 1 receptor silencing in vivo even. So if you injected it in the spinal cord, you could even find the effect of this calanin receptor antisense on pain transmission. I will not go into these details, but what I learned is that the synthesis of such covalent compounds is desirable, but it's quite difficult. So we were looking for non-covalent strategy for connection with CPPs of oligonucleotides, and it showed that our peptide series, when co-incubated with oligonucleotides or plasmids or whatever was needed, they formed the nanoparticles, and these nanoparticles were also transported into the cells, and somehow they released the oligonucleotide, and you got fantastic effects with that. So peptide, which was sterylated, as you can see here, probably forms a kind of complex based on charged amino acids and sterylated part. And here is one proposal what this complex could look like, but it is not confirmed totally. Again, many unclear things. And here one of my students made this figure about the mechanism, so you can see how this may happen. But again, I say it's not confirmed 100%. We believe in this, and there is a lot of things to do. So far we did transport of different types of oligonucleotides. You can see here on this figure, and peptide 14 has been one very good substance which was introduced by Matthias Helbring. So peptide 14, here you can see some more data about it. It was siRNA result here to knock down a protein on the experimental animals in liver. You can see the clear knockdown. And we also did some work together with Erkki Ruoslahti to develop our peptides into the homing peptides, which could come to the correct place. I will not stop here. Everything is available in these books. Latest 3 are here, and you can find them in the web. And next one will be published in 1 year from now, approximately. It's already on the way. And there is I am in hurry. I show this. There is one drug available today which has been approved by NIH or FDA, sorry, which connects botulinum toxin with one of the such peptides, which forms a complex and which takes away your glabellar lines and probably you don't need the injection every week. You can have 1 week, you have an injection in every 2 months only, and it makes it much more cheaper and less toxic. So if you didn't know about this, anybody who's interested to get a smooth face, go ahead. It is approved. And just a few words about our company, which we called Glass Pearl, because we read the book, Glass Pearl game, Glass Bead game actually in original in English. And Lee is not here, but our 3 people are here, Tamas, Marko, and me. We formed this company quite recently, and we have our own patent, and I can reveal the principle because the patent is available in public domain today, that we have our special CPPs actually, which when we co-incubate with lipid nanoparticles, form nanoparticles, and when you want to take the oligonucleotides of different types into the cells, and even in vivo, then you can use this, and then it's much more efficient than only the peptides. So it's a kind of wonder formula. We are trying to find funding for this idea, and let's see if we succeed or not. And as I said, we have a transfection system for almost all kinds of oligonucleotides and some of the proteins, but this system can be developed. We don't know exact mechanism yet, and many labs are working on this, as we will see here today also. And thank you.
We have time for a quick question or a comment. Anybody wants to add here, ask?
Thank you very much, Ulu. With your startup, Glass Pearl, what kind of pathology do you target somehow?
Whatever I like and what I get money for. But we are aiming at the moment to Alzheimer's disease, which is the most difficult in vivo to get things to brain, and that's why it's most interesting also. And as you know, there are many targets, many proteins which you want to downregulate, and many which you want upregulate. And we can upregulate the things with mRNA, downregulate with siRNA, and we have also delivery for CRISPR-Cas system, which I asked for collaboration. They are not interested. They think it's so available for what they have already. So let's see.
So, and you just mentioned CPP for your strategy, for the strategy for Glass Pearl. So you mentioned CPP, but any kind of CPP? I mean,
no, we have a special mixture. If you are interested, It's in this patent application, actually, so I can give you the formula if you want to collaborate.
Okay, any other questions? If not, then thank you very much, Ülo. And we will continue with our program. So our next speaker is Priska Vaskera from University of Montpellier. She's a professor there. And she did her PhD in 2004 in the Free University of Berlin, Germany. And to cell-penetrating peptides, she moved to 2009 when she moved to University of Montpellier in France. And today her talk is Crossing Cellular Barriers, CPP Applications from Lab to Therapy. So please, Priska.
Thank you. Can you hear me? Okay, so it's my second time in Estonia, and it's always a pleasure to be here. And first of all, I want to thank the organizer to invite me to present my results. And secondly, I think we can say happy birthday to Ülo. It's not too late. I started my career also by synthesizing peptides by the solid phase synthesis. And in Berlin, in this beautiful, no, I will do it with my hand. In this beautiful building, we synthesize a peptide, and we use a special synthesize method, which is called a spot synthesis. And with a common peptide synthesis, you can do it on a cellulose sheet and screen there a lot of peptide libraries to develop maybe epitopes as vaccines for antibody, or also interfering peptide to control protein-protein interaction. And as a biochemist, I was more interested in the biological evaluation of these interfering peptides. And at this time, I found this wonderful book from Ülo, the Bible of the cell-penetrating peptide. And I select 22 cell-penetrating peptides of different origins, couple them with a fluorescent dye, and together with my first PhD student, we evaluate them in 4 different cell lines to have an idea and to have a better comparison of all the cell-penetrating peptide under the same condition. And as you can see, there are different internalization properties, low internalization, medium, or high internalization. And we look also on the chemical modification, for example, if the C-terminus of the cell-penetrating peptide plays a role in the internalization property, and sometimes no, and sometimes you see different pattern. So, and with this background, be able to synthesize interfering peptides, having a small knowledge on cell-penetrating peptides, I moved from Berlin to Montpellier to work more on a pathophysiological project, and this was the acute myocardial infarction. So this is the killer number 1 among cardiovascular disease, and you should know that in Europe, every 45 seconds, there is 1 heart attack. And this is a very medical urgent because the heart does not regenerate. So what happens in detail? So the heart attack is due to the occlusion of arteria, and to save the patient, you have to reopen these arteria to perfuse again the heart. However, there are some deleterious side effects. The abrupt return of the oxygen blood causes oxidative stress in the ischemic tissue, and this makes the protein ducts leaving the nucleus and interacting in the cytosol with the Fas receptor, and thereby activating the apoptosis cascade. And there is today no pharmacological treatment against this reperfusion injury. So with the spot synthesis, I was able to take the primary sequence of this DAX peptide, dissect them in overlapping peptides, synthesize it on a cellulose membrane, incubate it with the Fas receptor, and define an epitope, meaning the minimal sequence which could interact with the Fas receptor. And this DAX peptide was coupled to the cell-penetrating TAT, as I was at this time in the lab of Bernard Lebleu. We test also other cell-penetrating peptide, like the RXR or the PIP family, because at this time Bernard works a lot with Mike Gate. But the TAT peptide is the more efficient for this application. So with this conjugated peptide, we should observe the internalization in the cell, and this is what we saw. These are primary cardiomyocytes from neonatal mice, and you see a very nice internalization. But if the peptide should be in the cells, then it should displace or block the interaction with the DAX protein. And this is what we saw here in a cell assay, where we artificially induce an apoptotic stress with a staurosporine, and then after 6 hours we replace the medium with normal medium to make such a recovery, and we can see a dose-dependent reduction of apoptosis, which is measured by DNA fragmentation. In collaboration with Stéphanie Barrère-Lemaire, we evaluate this peptide in vivo. So she has in her lab a model of ischemia reperfusion injury in the mice. So we were able to make on a coronal vessel of the heart a little knot to make the ischemia. And after 40 minutes, the knot is released to mimic the reperfusion. And 5 minutes before the reperfusion, we inject our peptide in a dose-dependent manner. And we can clearly see that the peptide can reduce the infrac size, which is not shown with the peptide TAT alone. And this reduction in infrac size is correlated with a reduction of apoptosis, which is also reduced in a concentration-dependent manner. Then we wanted to evaluate if this peptide has also an effect on a more long period, because often when you have a heart attack and if you recover from this heart attack, sometimes chronic diseases, heart diseases occurs like heart failure and so on over the time. So we inject the peptide at the reperfusion, and then we leave the mice 6 months in our laboratory, and every month we perform an echocardiography to evaluate the injection fraction. The injection fraction is the volume that was ejected by your heart when it's worked normally, and it's a clinical index for the cardiac function. And as you can see here, we have an improve of the injection fraction from the non-treated mice to the treated mice. And after 6 months, the mice were sacrificed, and we evaluate the fibrosis, because you should know that each cardiac cells which die is replaced by a fibroblast. And this has an impact on the contraction of the heart. And you can see here that with the mice treated with our peptide, we have a reduction of the fibrosis formation of more than 50%. And as we have a dream as ULO that we should go to a therapeutic application, we have founded a startup, and this startup will work on the proof of concept of this cardioprotective peptide in a large animal model. So in this case, it is a pig. And we work together with the Medizinische Universität in Vienna. And after this, we will go further to the regulatory safety, which is necessary to obtain the authorization to make the first in-human clinical trial. The second part of our work is the work on the peptide-based nanoparticles, which are made of cell-penetrating peptide, which is called VRAP, for tryptophan and arginine-rich amphipathic peptide. And this is the work that we perform together with Carrie Diaconaté, Sébastien Deshayes, and Eric Vives. These peptides are composed only of 3 amino acids, So it's not so complicated in the synthesis and not so expensive because you have only 3 amino acids. And this peptide could self-assemble with nucleic acid to form a nanoparticle. We prove this by different techniques. We play around with the ratio between the peptide and the sRNA and see that at a defined ratio up to 10, we see that the sRNA is no more visible, it's encapsulated. So meaning there is a complex formation between both molecules. Then we measure the size of these molecules or these complexes that are formed, and we can show that it's indeed a nanoparticle because the size is around 80 nanometer. And then by electronic microscopy, we see also that this nanoparticle have indeed a round or globular shape. The internalization mechanism. Ulo said in this presentation that this is something that all people wanted to know, but there are a few ideas, but it's very difficult to prove that this is one route on another route. So there are a lot of endocytosis inhibitor which can be used. And here you show a co-incubation of our nanoparticle and the endocytosis inhibitor. So this is the one, it's a silencing of luciferase in cells, in glioblastoma cells. Here you can see the nanoparticle alone, and here with all the inhibitors, and you see more or less we have the same results everywhere. So if we block the endocytosis, the nanoparticle is still able to internalize the cells. And then we use some special cell lines, which are triple KO for the dynamine, meaning that it's an inducible triple KO cells, meaning that for a short time we can inhibit also all the endocytosis-dependent or dynamine-dependent endocytosis. And we perform 2 different incubation, one with transferrin, so this is the wild-type cells. So the transferrin is internalized, our nanoparticle is also internalized, and when we perform transferrin with both nanoparticles, we see a colocalization. But if we use the triple KO cells, the transferrin is no more able to internalize, only our nanoparticle are internalized. Then, together with Magus, he measured the internalization of the nanoparticle, which are gold-labeled in glioblastoma cells, and you can see here the interaction at the membrane, and here the nanoparticles in the cells. So, based on these results, we hypothesize that our nanoparticle internalized via direct translocation. Then we play around with our nanoparticle by adding different grafting entities. So first of all, we graft our nanoparticle with a PEG moiety. This was done to allow the transfection of the whole embryo zebrafish. So we use zebrafish which are genetically modified. They overexpress green fluorescence protein in their endothelial cells so that the vessels are labeled in green. And when we, by a simpling bathing method, incubate the zebrafish with our nanoparticles, which encapsulate an sRNA against the GFP, we can see a very nice knockdown. So the nanoparticle is able to go through the skin of the zebrafish and knock down the GFP. The second is the addition of a mitochondrial targeting sequence. So this is done in collaboration with Diana Costa in Portugal, because she's working on different mitochondria pathology. And here I show you one example. The plasmid is a fluorescent label, and when we use the naked nanoparticle, we have the plasmid in the cytosol, and when we have the MTS, the mitochondrial targeting sequence added, then we found the plasmid in the mitochondria. And interestingly, she just published, I think, last year, a paper showing that the internalization of the nanoparticle is dependent on the circadian clock of the cells. So it's dependent on which cycle of the cells you have a better or a lower internalization. And ongoing work is on the multigrafted nanoparticles. Here is an example of pegylation and a targeting sequence, which is a cyclid RGD, to target cancer cell line overexpressing beta integrin receptors. And you can see here that if you have a low grafting percentage, you have a good knockdown. But if you increase the pegylation to 100%, then you lose the activity, maybe or probably because the pegylation will mask the nanoparticle and avoid the internalization. And therefore, we collaborate now with Sebastian Ulrich, and he has several pH-sensitive linkers which were added between the PEG moiety and the VRAP peptide, so that the nanoparticle is decapsulated, depegylated at the tumor size where we have a pH drop. And here again, a more physiopathological application. We work a lot on the cystic fibrosis. As you know, the cystic fibrosis is an autosomal recessive genetic disorder. It affects the lung, but also other organs like the pancreas, the liver, or the intestine. And this pathology is due to a mutation of a chloride channel called CFTR. And this mutation causes different defects. First of all, the CFTR is not properly folded, so it is rapidly degraded by the proteasome. However, there are a company, Vertex Pharmaceuticals, who has developed correctors. These are small molecules which help the CFTR to properly fold and to reach the apical membrane of endothelial cells. Epithelial cells, sorry. And once at the membrane, the open probability of the receptor is not very good, but here again, there is the same company, Vertex Pharmaceuticals, has developed potentiators which increase the open probability of the receptor so that we have a higher chloride efflux. What we also know is that the amount of endogenous CFTR transcript, so meaning mRNA, is very low in the lung of adults compared in the lung of fetal. There is a switch between the babies to the adults. We lose the expression of the CFTR. Also in non-CF patients. So meaning that if you have a low CFTR mRNA quantity, you will have also a low protein quantity. So in healthy tissue, you have a good chloride efflux through the channel. The mucus is very fluid and could be transported by the cilia cells. In the case of cystic fibrosis, we have some mRNAs which target the 3' UTR region of the mRNA, and this blocks or induces the mRNA degradation. And if the mRNA is degraded, you have less channel activity at the membrane. So what we can do is to use some small oligonucleotides called target site blockers, which interacts also with the 3' UTR region of the mRNA, and they block the interaction of the miRNA. So if the interaction of the miRNA is blocked, so the mRNA is not degraded, and we have again, or should have, more CFTR at the apical membrane. And this work is done in collaboration with Magali Tolonka-Dars. She has proved this mechanism on 3D nasal epithelial cells issued from patient, because for this pathology we have no in vivo model representing the real symptom in humans. So the assay are always done on on cells from CF patients which are cultivated in the laboratory. So Magali shows that we have, when we treat the cell with a TSB, an increase in mRNA level, an increase in the protein level, and more importantly, also an increase of around 50% of the channel activity. So you will ask me why we need a cell-penetrating peptide in this context. Because these TSB, these small oligonucleotides, are known to accumulate in the liver and to cause hepatotoxicities. So our idea was to encapsulate the TSB with our nanoparticles, RAP-based nanoparticles, to reduce the concentration or to have a better targeting. And this is what we have done. So like for the sRNA, we have formulated some nanoparticles at the defined molar ratio. Though this is the molar ratio between the peptide and the TSB of 5. And we can measure a very nice size of around 60 nanometer of the nanoparticle. And more importantly, they are stable at 4 degrees for over 30 days. Then we looked on if this nanoparticle can be stored as a powder and lyophilized our nanoparticle and resolubilized, and checked if we always have this nanoparticle or if this freezing reconstitution process will engage the formation of the nanoparticle. And as you can show here, if we use different cryoprotectants like glucose or mannitol, we have also nice nanoparticle after the lyophilization. And today we are working with microfluidic systems to improve the formulation so that we really have nanoparticles which have all the same size. Then we prove the efficacy of the nanoparticles. So here we work on bronchial epithelium from CF patient, and we can see that with our TSB nanoparticles, we increase the amount of CFTR mRNA. It's the same amount like the TSB alone. But when we look on the activity, we can improve the activity to 80%, which is higher than the TSB alone, which was previously around 50%. But more importantly, we show some difference in the biodistribution of the TSB alone versus the TSB encapsulated in nanoparticles. We administrated it via the intranasal administration. And after several times, we look on the fluorescence, so the TSB is fluorescent labeled. We look on the fluorescence in different organs. And for the TSB alone, we can see an accumulation in the lung, for sure, but also in the kidney, the spleen, and the liver. But with our nanoparticle, the accumulation is still in the lung, maybe a little bit higher, but we have less accumulation in the other organs. And when we look the lung in detail, we can see the accumulation here in magenta, the TSB in the epithelial cells of the lung. So, and here again, these results were so nice so that we decided to make another startup focused on the cystic fibrosis. And with this startup, the objective will be to test these nanoparticles on more patients with cystic fibrosis with different mutation, and then also to go to the first clinical trial in humans. So I think I can continue like, or I hope I can continue like Ulo, meaning that I could show the development of something from the lab to a therapeutic application, I hope in the near future. As Ulo, we need some money to develop everything, and this is today a little bit problematic because with the situation, the worldwide situation, the investigation in young companies are not so good. But we don't give up and hope that it will reach once of this project, the clinical trial. So at the end, I want to thank the group where I work, the different people involved, our collaborators over the time, and for sure all the institution for the financial and you for your attention.
Thank you. It was very nice. After international administration, you didn't show brain distribution.
Yes.
Too good or too bad?
Indeed. So we have systems that the mice is not on the horizontal, but it's a little bit decline in this position when we administrate. But the final idea will be to have oral application or an aspiration for the patient. And in the mice, then we will mimic this with an intratracheal administration.
Yes.
Thank you. Very nice talk. I have a question related to the RAP nanoparticles, because usually when you work with nanoparticles, you have somehow when you inject the nanoparticles in the circulation, the blood circulation, you have the corona protein working on. So what is your experience with this corona protein?
Yes, it's true. So mainly when we go in vivo, we have always a small percentage of pegylation. This will not reduce totally the interaction of the corona protein, but reduce it. So we have performed some assay in mice and measure then the, determine the sRNA was, so the nanoparticle encapsulate a fluorescent labeled sRNA, and we inject different nanoparticle versions, the naked, 10%, 20%, 100%. And we can see that if you add more and more pegylation, you will have a circulation of the fluorescent sRNA longer over time. However, if you pegylate too much, then it will not internalize anymore. So.
Hey, my name is Anna, and I also work with Ülos Peptide. And I have more biological questions. So it was very interesting approach for the cystic fibrosis. And my question is actually, so in which mutations, in which case it works actually? So is it also the common delta 508 mutation?
Yeah, we have tested this, our nanoparticle on homozygote delta 508 mutation on heterozygote. And then you can normally also target other mutation by simply switching the TSB. So for example, we are working
on
also...
the 3' UTR, but somewhere else than binding or...
No, we are, yes, we are also working on non-stop mutation, on stop mutation, and we use the TSB to mask the stop, so that...
okay,
so,
that makes sense.
Yeah.
Yeah. Okay, thank you.
Any other questions? Yeah, Tamas.
You have ended on a very provocative note, saying the bird is such that it's very difficult now. But I think this is a rather, I am sure that you are aware of that when we sit in this room and we look at the people who have brought this to medicine, they worked on cholinesterase inhibitors during the Cold War on both sides of the camps, and they did do the best to develop scientific concepts. And they did as much. You cannot lift out your project of the world we are in. The people who worked on cholinesterase inhibitors and organophosphates were not only thinking of pesticides, whether they worked at Mill Hill in the United Kingdom or Porton-Dunn in the United States or everywhere in the Soviet Union. And this, we have to see that good scientists will be able to do the best of abstraction of concepts and keep up a hope that those concepts then can be used in your case therapeutic. The Hungarians taught me, the Hungarian Nobel laureate who remained during the Second World War in Hungary and who was almost shot by the Soviet troops when they arrived because he distributed the food which was for them, was Albert Szent-Györgyi. And you know what he worked on? The only thing in Hungary you could get grants for was paprika. He not only discovered vitamin C, but he closed the Krebs cycle. Krebs cycle became cycle. I used to share a room with them at Woods Hall. However, the newly dependent Hungary in 1946 did not want him as a president. The Peasant Party sent a person and told him, Professor Szent-Györgyi, you better leave Hungary. We don't want people like you. So, you know, I think we cannot dream of that we don't live in a real world. I think we teach all of our students scientific enthusiasm, abstraction of scientific concepts, quantification, and try to publish everything, absolutely everything, and let the reader learn so much as they can. And those who pretend that this is something new, there is nothing new. Unfortunately, nothing new, and also therefore the aims cannot change. I am sorry for this, but it's better to do it at the end of a beautiful lecture because you built on exactly this, that you picked up the things which had to do with concepts which were useful for your work to go from preclinical experiments in biochemistry to therapeutic approaches in rare autosomal diseases. When we founded Vertex, we had a goal to work on cystic fibrosis, and we had no idea about the roots.
Yeah.
And now there are more than 18 different approaches. And there are 3 approved drugs. They are not safe, they are not perfect, they don't work on every mutation. But I would maintain that the world has not gotten worse or better. It's rotten. But scientists can, for times, work together and above.
Yeah, this is important.
Thank you very much.
Yeah, and maybe I will also only close a short closing remark is, so for 2 years ago, I organized in Montpellier a cell-penetrating peptide symposium. Unfortunately, you couldn't come, but yeah, who's the next?
So our last speaker of this session, or the morning session, is Professor Margus Pooga from Tartu University, from Faculty of Science and Technology, where he's a professor of chemical biology. And Margus got his PhD in '89 under supervision of Ülo Langel. So please, Margus.
Thanks for the privilege to speak about CPPs in front of so many people. And I titled my talk From a Chimeric Peptide to Nanoparticles, just to emphasize the role of Ülo in this journey. When I was invited to Stockholm by Ülo and Tamas, one of the main goals was to purify galanin receptors, which were not sequenced yet or even not well known. Ülo designed a peptide that consisted of consisted of N-terminal part of galanin that had high affinity to its receptors, and mastoparin that was known to bind tightly to membranes. They were connected by lysine that was biotinylated. So with this three functional chimeric peptide, we hope to pull out galanin receptor from solubilized membranes of big brain. Actually, we did not succeed with it, despite of lots of efforts from different people in this room and enormous resources. But it appeared to be quite specific. The physiological effects of this peptide were very weird, not mediated by galanin receptors, and quite at the same time, cell-penetrating peptides were discovered by Alain Brachan's group, and the effects of this peptide were more from inside cells, not from outside. So we incubated cells with this peptide and later detected where it is. It was not classical binding to receptors at all. It accumulated inside cells. And the pattern of localization resembled to membranous localization of membranes. So by combining something from brain and toxin from wasp, we got a peptide that was able to dissolve both in water and in hydrophobic interior, like inside membranes, and was switching the conformation upon this transition. Ülo and it already briefly touched it. If you have a peptide that can internalize, you can deliver inside cells whatever. Here it is avidin that is targeted into cells in red, and quite efficiently in 2 hours already. But then we went down with temperature. The peptide did not deliver protein anymore, so it was for larger particles, perhaps direct penetration is inefficient. We could also deliver analog of DNA called PNA, was favored for Ülos group for quite many years. And in the beginning of this century, optimists said that everything is possible to deliver into cells with CPPs, either by strong force or rocketing anything inside cells, like trends in pharmacological sciences showed in their cover. Science was fun, and even peptide synthesis was fun. That is quite a tedious thing here. I acknowledge younger colleague who passed too early. Okay. I was still very much interested how actually CPPs get into cells, and I happened to be in a department that had some expertise in electron microscopy. So we labeled a CPP with a small gold particle to see each and every peptide on the cell surface. So here, every black dot shows one peptide. For our surprise, the peptide clustered into some particles by itself, and these clusters were able to directly translocate inside cells. When we used higher concentrations, these particles were higher, larger, and they were quite well structured, showing that perhaps we have multilamellar particles. If you talk about mechanism, I think these pictures are quite well in line with the hypothesis of de Hoppe, who thinks that peptides, including CPPs, are mostly internalized by endocytosis, and endosomes bud, giving smaller vesicle that just disrupts. Perfects, I think, were the major breakthrough in delivery of nucleic acids inside cells. Perfect 14 was already mentioned, and I don't speak long here, but here splicing of introduced gene is rescued with splicing switching oligo that complementary binds to aberrant splicing site, and luciferase activity is restored inside cells. This has been one of favored models in our lab and in many others. It was pioneered by Bernard Lebleu's lab, I think, in field. Perfect 14 is quite an interesting peptide by itself, and it has really bad reputation in many labs because often the results do not reproduce well, and the in vivo toxicity is often met. We tried to understand it and again used electron microscopy. So if to dissolve perfect 14 in water, it often shows aggregates. If to sonicate this solution, particles start to form, but they are not very regular. But if to dissolve the peptide in organic solvent and then dilute in water, you will have nice particles depending on the composition of the solvent. strangely, if you dilute this peptide stock not in water but in herpes buffer, it starts to form elongated micelles. And with some tricks, we can get FF14 as structured nanoparticles that perhaps are more easy to disassemble inside cells. As it was told earlier, endosomal escape is the limiting step for all nanoparticle drugs, including CPPs. We have tried to find ways to increase the efficacy of endosomal escape or just make particles with FF14 and nucleic acid more efficient. First things that we tried were calcium ions because they can associate tightly with delivered nucleic acids and work from inside cells. And indeed, my graduate student Maria showed that if to include calcium or magnesium inside particles of CPP with nucleic acid, the biological effect grows quite substantially. And now we have several other compounds that increase the efficacy of CPP nucleic acid nanoparticles. CPPs work extremely well in cell culture. What about in vivo? Fortunately, I had a good colleague in next house, Professor Anna Rebane, in medical faculty, and in her group, we started with delivery of microRNAs. The model that we used was irritant contact dermatitis because of its simplicity and quickness. So we packed anti-inflammatory microRNA 146A into particles, and this microRNA suppresses expression or signal NF-kappa B. Shortly, the model is that inflammation is induced in air, and we try to counteract it. And the contact dermatitis is probably known to quite many people as contact-induced dermatitis or just inflammation of skin. We used PEFECT6, that was the best vector in these times. And the particles formed nicely. They readily entered primary keratinocytes that usually quite reluctant for transfection. And by subcutaneous injection, we introduced it into mice. And by this dye 547 dye, we estimated their or analyzed their localization. For our surprise, in normal tissue, the particles did not spread much. But after inducing inflammation, the spread of microRNA was much larger. If to quantify with other small dose of microRNA, we were, or Anna's group was, able to substantially reduce the air sickness and inflammation, and also the NF-kappa B pathway responding genes were downregulated quite well. Perfect-14 is really good, but as chemist, I tried to make more efficient peptides compared to original Perfect-14 that was designed by Halbrink, as it was said already. The second series was designed by Lee Porosk, former graduate student of ULO, and as I understood, co-owner of company. In first series, we learned that the helical structure of this particular peptide is clearly necessary. If we modify either hydrophobic or cationic phase of it, we usually lose the activity. In principle, there are several ways what we can do to make peptide more efficient. One possibility is to cluster all charges in one place, reduce positive charge, play with hydrophobicity, and introduce amino acids that could escape at endosomal escape. And this is not just simple trial and error, at least predicting the properties of peptides, and has been really successful in my understanding. From microRNA, we switched to messenger RNA. With oligos, CPPs work very well. With mRNA, it is much more difficult, but luckily, we got peptides that are more efficient than Perfect-14 itself, or it is analog with lysines. And more efficient are peptides that are a bit more hydrophilic for mRNA delivery, I mean, and which could promote escape from endosomes. Oh, okay. Yep. And this we studied in Hakat cells, a proxy line for keratinocytes to be sure that we can work with skin delivery also. Yeah, I forgot to mention that Anna's main interest are inflammatory diseases with immune background, and she is mostly using skin models and lung models. So, in cell culture, CPPs work really well, including primary keratinocytes. At the moment, we have several peptides which are more efficient in delivering messenger RNA and expression of reporter protein compared to Perfect-14, and they are more efficient than lipid carriers also. Sorry. We were struggling long with in vivo delivery of mRNA by subcutaneous injection. And yeah, we were misled by good results from cell culture experiments, so we had quite profoundly to change the formulation of these particles. But we were happy to see that with Perfect-14 it is possible to get expression from luciferase mRNA in mouse skin. And when we make hybrid nanoparticles, we can gain with this expression. And these experiments were done in Professor Rebane's lab by two very enthusiastic doctoral students. So, to conclude, we think that perfect type CPPs form micelles by themselves. These form larger conglomerates or coacervates in chemical means, and nucleic acid is packed somewhere between or on its surface. And the biological efficacy of such nanoparticles can be substantially increased, adding different excipients into these nanoparticles. And since there is a hydrophobic core, positively charged and negatively charged parts, almost or quite many different compounds can be well encapsulated inside these particles. And we believe that CPP-based nanoparticles are less immunogenic compared to regular lipid nanoparticles that are commonly used for transfecting mRNA into cells. And first, I want to thank Alain Prochain for kicking off this field with a quite bold hypothesis that peptides and proteins can travel between the cells, and Ulo for pushing the field forward for several decades. Of course, I thank Anna and her group and my groups from different times of my own lab, and also department who helped me in electron microscopy in way or another, and you for your kind attention.
Thank you very much. Do we have questions? Yes.
Dear Margus, you said, just straight without any specifications, that your CPPs are less immunogenic than lipid nanoparticles. But if you can truly prove it, you know, lipid nanoparticles with the COVID vaccination made the world. And if you truly can prove this, then you have a big future for your specific. So should you say CPPs with this and this charge, what you include into them? Or does it depend on what you carry in? Because, you know, the whole business of mRNA vaccines with somehow helped us through COVID was based on that lipid nanoparticles came in, and they came at a very high cost of side effects. The first group of American Navy recruits, as you know, had to be put on shore somewhere, unknown destination for months, because they had heart inflammation. The second group was another Navy group from the Israelis. They had severe autoimmune problems, which no cortisol treatment, and very difficult effects remained, and we don't know how long. So can you just say this, or you have to qualify it depending on what is the cargo, or it's independent of the cargo? If it is so, then can you try to do what has been now in millions of people, the COVID vaccine? We know the sequence, we know everything about it. I can get it from you from Pfizer, from BioNTech. You can get a ton if you can get the CPP to wrap it.
Yeah, absolutely sure that if to talk about the immunogenicity, we have to take case by case, one partner, another partner, and the mRNA by itself is very immunogenic.
That's uninteresting. Nobody wants to deliver a delivery vehicle. You want to deliver it because you want to deliver the cargo.
Yep.
Is it true with every cargo?
I don't think so. I am.
Think so? You don't know.
I know only partly. I don't know.
You know it for an important cargo. Because this would be the heart of the matter. To find an important cargo where CPP indeed is less immunogenic than LNP.
Yep.
After that, we can discuss how much they cost, and all that changes once you start to make millions of doses.
Yep. And why I tried to say it was that we were interested in chronic inflammatory diseases and suppression of them. So yeah, for kind of vaccine approach, we haven't done anything. But in cytokine release,
Given to as many people as a vaccine.
Yeah.
It is not a single medicine, not even aspirin, given to so many people as a good vaccine.
we are just in the beginning of our path.
It's worth
Yeah.
doing the experiment.
Yep, sure.
Your doctor can give you the dose from any of the used COVID vaccines. You can take away the LNP, you know how to do it. You can wrap your CPP, come back and tell us.
Yeah, next time.
That would be the big deal.
Yeah.
Okay, thank you. Any other questions? Intriguing comments? Yes.
Thank you, Margus. Very interesting talk. I have a question related to PIPFACT 14, because you mentioned that it was terrible to work with it because it's a chameleon-like behavior concerning, I mean, according to the conditions you use, the experimental conditions you use. So my question is, for in vivo delivery, do you have any idea in which form is the PIPFACT 14 active?
We are not interested in systemic delivery because of protein corona and very wide distribution as it was shown more than 10 years ago, I think. We rely more on local injection for some conditions. And there, yes, I think we start to understand what we were doing wrong for years. So for locally, yes, this is again a question that I can answer in about 2 years.
Okay, Okay, so I think we can then continue the discussions over the break. So we will now have a little break, but we thank again all the speakers. And we meet here at 2 o'clock, so almost an hour break. And then the food is served there, so we can open that door. So there are tables there, but there are also tables there in the lobby, so you can take your food there if not everybody fits there. So see you at 2 here. Thank you.
Okay, we will start with the second half of our conference. I think we are still waiting for some people who are missing. My name is Liiv Illo. I'm here representing Estonian Biochemical Society. And I will guide you through the second half of our conference. And our first speaker in this afternoon is Sandrine Sagan. Professor Sagan is from Sorbonne University, and the rest of her affiliations are so long that it takes too much time to read. So I will just introduce shortly that her work has significantly advanced our understanding of how cell-penetrating peptides cross biological membranes. And now she will give an overview of these molecular mechanisms to us. Thank you.
Thank you very much to the organizers to invite me to have the opportunity to give a small journey about cell penetration and regarding more particularly translocation, which is a very interesting way to internalize into cells. And I would like to start to say a few words about Ulo. I'm really very happy to be here today because you're a scientist who can serve as a model for young people. Yeah. Because you're a very good scientist, brilliant, talented. But not only that, you are very modest and you're very honest. And I think we need such kind of scientists in science in general and in the field. So, see? And you know, I always appreciated your sense of humor. And there is a French writer, Alphonse Allais. I don't know who knows it, who knows him. Alphonse Allais, and he said something like, I will translate, of course. He said that we don't have to take ourselves seriously because at the end there will be no survivors. And I think you're really this kind of people making very serious things, science, but you don't take yourself very seriously. So this is, thank you, Ulo, for gathering all over the world people with meetings and your books, as it was mentioned this morning. And just before starting, I give you a small present from So
Now back to science. I will start with the origin of the field, as mentioned this morning. So this was the group of Alain Prochian in Paris, together with Alain Joliot. So they showed first in this paper from 1991 that the polypeptide derived from the Antipenapedia homeoprotein could indeed internalize into cells. And this was the beginning of the story, because a few years afterwards, so they could show that making these peptides from the helix 3 of this polypeptide, so they could identify that the sequence 43 to 58 so was indeed able by itself to internalize into cells. And the remarkable thing is that they did the experiment also at 4 degrees. And again, the peptide was able to internalize into cells. So this was the start of the field. And in the team, we have been involved since 20 years in the field, and our aim was really to try to understand the cell penetration by a non-toxic membrane, local perturbation events, the so-called translocation. So what we did first was to be able to quantify how much peptides could internalize into cells. So we developed over the years different methods to be able to quantify peptides into cells. So it was quantification by mass spectrometry, quantification by direct fluorometry, and more recently quantification by after Fred quenching. And I will present some aspects of these methods. So the quantification relies on the use of a non-deuterated version of the CPP, which was labeled with biotin and glycine, and also another peptide analog, which contained bideuterated glycine. And of course, the two peptides have a mass difference of 8 units. And what we did is that after incubation of the cells, so 1 million cells with a concentration of the non-deuterated version of the peptide, and by incubating these cells either at 37 degrees, so meaning we could identify both endocytosis and translocation, or 4 degrees with only direct translocation. And after incubation, washings, and of course, because these peptides are really membranotropic, you have to get rid of the membrane-bound peptides. So an accurate peptide degradation to remove the membrane-binding peptides, and so to keep only the internalized peptides. And afterwards, during the cell lysis, we introduce a known amount of the bideuterated version of the same peptide. So meaning that in the mass spectrometry, the peptides behave exactly the same, I mean the same ionization ability. And because thanks to the biotin labeled peptide here, we could fish out, we could purify the two peptides out of the cell lysis using streptavidin-coated magnetic bits. And, it seems that the mass spectra disappeared here. Yeah, so sorry, so this is, so you should see an isotopic massif here. So from the non-labeled peptide here and the labeled peptide here, and because we knew how much of the bideuterated peptide we introduced, we could get direct access to the internalized
quantity of the peptide. So this was done also a few years afterwards using fluorometry. So we applied the same. So we developed a method to using, oops, sorry, is it correct? No, sorry. So it's moving here, I don't know why. We used fluorescent label peptide because when you're using imaging, for instance, fluorescent imaging, of course, you see the fluorescent peptide, or you see at least the fluorescence, but you don't know whether the fluorescence you are watching is still attached to the peptide. So this is the first thing. And the second thing is that you may have some quench processes when the peptide is for instance, accumulated in high amounts in cells or within the membrane of cells. So to be really sure about, to be really sure about how much peptide we have inside cells, so we proceed again to cell lysis, and again by using a fluorescent standard curve, we can have access to the internalized peptides inside cells. And we developed both methods, and we could show with one single peptide that is penetrating here, that when we use, that was labeled with the biotin or labeled with a fluorescence probe, that by using the two different methods, so either mass spectrometry or fluorescence spectroscopy, we could determine exactly the same amount of peptide in cells. So showing that the two methods are really robust because they are really a very different way to address the quantity of peptides inside cells. And more recently, what we used was to develop a Fred quenching and release approach to evaluate translocation in cells. And the way we did it is that we suppose that when the peptide internalizes into cells through endocytosis, of course, it ends up somehow in endosomes. And of course, you know that there are enzymes in the lysosomes. And so that the peptide can be cleaved. And then when the peptide is cleaved, of course, you have no longer quenching of the fluorescent probe, and then you can detect the fluorescent signal. But when the peptide internalizes into cells by translocation, the peptide is not degraded, at least during the kinetics we were using. And of course, you cannot detect the peptide here by quantification. And so if you incubate cells with this type of peptides, so you have both endocytosis and translocation inside cells. If you boil the cells afterwards, it means that you totally inhibit enzymatic degradation. So you only keep the peptide cleaved that has been cleaved during endocytosis, and the peptide that has been entering through translocation is still intact. So you don't detect it this way. So you have a way of quantification of only endocytosis. And alternatively, so you double the samples, and if after incubation with the peptides, you do a lysis of cells, of course, you release also the cellular enzymes. So everything inside cells is degraded, and there you have access both to translocation and endocytosis. And if you subtract, of course, the two conditions, then you have access to what has been translocated. And this is very important because then you can measure translocation at 37 degrees and no longer at 4 degrees as we did previously with the previous method. So this is a very robust method as well. And what we did is what we show, for instance, so here you have,
we introduced the dapsil as the quencher of the carboxyfluorescein here on the R4 peptides or on the R6 peptides. And for a comparison, we used also R9, which is a very well-known CPP, and not containing dapsil, just also to evaluate the introduction of dapsil, so this hydrophobic group, into the oligoarginine peptides. And so we used the protocol I just described. And what we could show is that, so you have in pink the translocation process, which has been determined through this approach, and the endocytosis. And you see here that very interestingly, the R6 peptide internalize in an equivalent way through translocation and endocytosis. And a very interesting point also is that, for instance, R4, but which contains a dapsil group in comparison to the R9 here, internalized through translocation in a similar way as R9. So showing that we could use also this method to determine which paths are taking the CPPs. So how can, so we used now, I will not go into details on how we quantified the peptides, so using one of the methods I just described. So of course, the questions are how can a positively charged polar peptide cross a hydrophobic barrier? So, of course, it has been described in particular by the group of Paul Vender in the United States that the arginine groups are very interesting amino acids because they can form very strong H bonds through bidentate H bonds, in particular with sulfate, carboxylate, or phosphate groups that you found at the surface of the, well, within the membrane or at the surface of the membrane within a phospholipids group, for example. And what we found, and not only us, but also other groups, is that tryptophan is a very important amino acid. Not only because it's a hydrophobic amino acids, because, for instance, if you take the penetrating sequence, which contains 2 tryptophan in the sequence, if you remove one or the other tryptophan from the sequence by replacing it, for instance, by phenylalanine, which is a very close amino acids in terms of hydrophobicity, sorry, you lose totally the internalization capacity of the peptide. So, meaning that tryptophan has something in addition to, for instance, phenylalanine or tyrosine. And what we discovered is, for instance, well, this is just to illustrate as well, is, for instance, we used nonapeptides, so composed of only arginine and leucine, or arginine and tryptophans. And what we recognize from differential scanning calorimetry is that one peptide, so LR9, was able to insert more deeply in the lipid bilayer compared to tryptophan, which stayed more at the interfacial leaflets of the lipid bilayer. So meaning, and the very interesting point is that RW9 is a very good CPP in terms of internalization efficacy, and RL9 is not a CPP. I mean, it internalizes very hardly in cells. And so it shows that somehow hydrophobicity is not the only parameter in the activity of tryptophan. And what with a very nice collaboration with Antonio Frontera, who is and his colleague Antonio Boza, who are chemical, theoreticians, they showed that using DFT calculations
and using, using, so this is a sequence of RWWR and RGGR sequence, peptide sequence, and they introduce in the system, in their calculation, a small fragment of glycosaminoglycans, so monosulfated glycosaminoglycans, and what they show very nicely is that there is a salt, there is a salt bridge P interactions able to form between arginine and tryptophan-rich peptides compared to only arginine peptides. And these ion pair P interactions do enhance the energy of binding between the sugar and the peptides. So meaning that, and tryptophan is the only amino acid which is able to do that. And meaning that, so we have a lot of so-called weak interactions that can go into motions between these peptides and some partners at the cell membrane. So the guanidinium moiety is able, of course, cation P interactions. Of course, when you have also tryptophan in the sequence, you can have electrostatic ion pair interactions, and also ion pair P interactions, and of course hydrogen bonds. And you see that tryptophan in parallel can have many different interactions that really are very important, at least for the peptide interactions with the components of the lipid bilayer, and afterwards for the translocation, since, of course, the interaction is the first step before translocation. So, as the DFT models show that we can have ion pair P interactions, we addressed, well, since many years, the role of GAGs in CPP internalization. And what we showed is that, of course, all the peptides, if you take one concentration and one cell type, the different CPPs internalize very differently one from the other. And this is also true, this is true at 37 degrees as shown here, but also at 4 degrees. And if you use a different cell line, which is deficient in heparan sulfate and chondroitin sulfate, you see that this is still the case. So the peptides internalize again quite differently. I have to mention at that point that these CHO cells that are deficient still have about 5 to 10% of GAG at the cell surface. So they are not nude in terms of glycosaminoglycans, which is quite important. And so, what is the role of GAGs in the vicinity of the lipid bilayer? So, if you take pictures that you are schematic representation of what is a cell membrane, very often you see this kind of images. So, meaning that GAGs seems to float above, pointing towards the extracellular milieu compared to the lipid bilayer. But what we could show is that it's not only that, because again we use calorimetry, differential scanning calorimetry, and we could show here, for instance, we take heparin as a model for GAGs at the cell surface. And so we use vesicles, loofs of DMPC, composed of DMPC. Oh, come on. I'm sorry. I'm not familiar with this. And what we found is that heparin, so you see here, in the absence of calcium, heparin did not interact. I mean, it did not move the thermotropic profile transition phase of the MPC. But as long as you have calcium in the buffer, you see here that the present transition temperature disappear, and we have a modification in the enthalpy and the transition temperature of the MPC, showing that heparin in the presence of calcium is able to bind phospholipids. And we could show exactly the same with another very important GAX, which is the 4,6-sulfated chondroitin sulfate here. So our CSE is also the name. And it's also true, and it's even more pregnant. It means that the interaction is even better between CSE and the MPC vesicles. And again, it's totally depending on the presence of calcium in the buffer. And what we show afterwards is that, so we use this peptide, so I don't have time to present in details the peptide, but it's derived from the homeoprotein, OTX, that has been identified by the group of Alain Prochion as a GAG targeting domain in the homeoprotein. And so we synthesized this chimeric peptide containing this GAG targeting GAGs together with the CPP. And what we show is that the peptide could compete in terms of interaction, with CSE. So meaning that the GAGs are able at the cell surface to directly bind to the phospholipid bilayer, so being really in the proximity of the lipid bilayer. And this was evidenced more with the binding of CSE here by tryptophan fluorescence blue shift. I don't have time to go into details. So you know that there is a fluorescence blue shift of tryptophan when the environment of the tryptophan is changing. And what we could show is that, so you see here that the peptide has some quite low affinity for heparin sulfate, and as expected, has a better affinity for CSE. And when you remove calcium, the affinity doesn't change between the two. And if you use now decorated vesicles, so DOPC vesicles decorated with either heparin, you see that the peptide, there is no change in the tryptophan environment. But if you use CSE decorated DOPC, then you see a dramatic change of the affinity. So meaning that somehow the peptide do interact with and do insert within the lipid bilayer of the vesicles decorated. So, and this was corroborated in cells. So we use GAG deficient cells, and you see here that if you add extra, so these cells do not contain or very poorly glycosaminoglycans, and we add extracellularly CSE, and you see that only with the peptide, so these are other non-related peptides, so only this peptide could be enhanced in terms of internalization with this CSE addition, so corroborating somehow the hypothesis. And so this is the current vision of how we see things. So you see in syndecans, for instance, present in the cell membrane, the CS GAGs are present at the lower level of the syndecans, whether the HS heparan sulfate chains are upper, so more distant from the lipid bilayer. And so we could imagine that the peptides, once they recognize the binding, so they are at the close vicinity of the lipid bilayer, and then they can interact and insert through the tryptophan within the alkyl chains. And so this is now to finish a very recent work, which is not published yet, but I hope we could manage it. So we developed a double patch clamp electrophysiological experiments. And so what we did is that we have the cell attached pipette here. So it means that the cell membrane is intact, and you have the pipette, so making attached to the cell surface, whether the other pipette here is in the whole cell recording, so meaning that the pipette is inside the cells. So through this pipette, you can control the membrane potential, and through this pipette, we can add here the CPP or the homeoprotein, and of course, we can, in the same pipette, also detect current events if ever they occurred. And so we did this with several CPPs, as mentioned here, and we did it also with different cell lines and also brain slices to make sure that it is also biologically relevant. And so what we did, of course, we could not think about a channel, because a channel is something very stable. We could not think about antimicrobial pore formation, because pore formation through antimicrobial peptides is very stable as well, and of course it leads to the bacterial death. So we imagined first that we could just improve or enhance the noise, the current noise of a cell. So of course we had to be able to detect it. So and it was not possible by hand, so we developed a modelization system, so meaning that we could not by our hands, but through a computational calculation, we could detect. We could have a measure of, so the green line here is the average noise of cells, and afterwards, because of course this is noise, so you have Eckart type, standard Eckart to moiety. So of course we decided to detect events a bit upper this mean. So this is the red line, and of course what we did is that we analyzed 10 more than 10 points by 10 points, all the traces, the electrophysiological traces, and we decided that if we have more than 10 points upper, then here there is an event, and if not, it's in the noise of the cell. So using this automatic computational model, we could detect that yes, the CPPs can induce pore formation during translocation. Why is it translocation? Because the frequency and the duration of the pore formation is much more faster than any kinetics that have been described so far for endocytosis. So it's about 100 to 1,000-fold quicker than any endocytosis process. So, and all the peptides, the very interesting points is that we could detect events even at very low nanomolar concentration, and this is an idea that we are convinced about for many years, that translocation is first. So meaning that at low concentration, translocation is first. On the cytosis, you need some cooperativity, you need accumulation of the peptides at the cell surface, and so it started at higher concentration. And here we could detect events even at 10 nanomolar. And the very interesting point is that whatever the peptide, TAT, R9, penetratin, or the other, the frequency of pore formation and pore duration are similar, meaning that all peptides do induce pore formation the same way. And the very interesting point is also that here the non-penetrating peptide has induce no events at all. And I would like also to mention just the effect of temperature, because of course, the effect of temperature, here we could show that these events occur also at lower temperature, and the impact of the lower temperature is only on the frequency of opening, so it lasts between two events, the time is longer, but the opening duration again is very similar at 4 degrees. So just to make a long story, there is much more in the paper, but I don't have time to describe here. So translocation can indeed be assimilated to a transient pore formation. And what we show in the paper is that it's the same for homeoproteins. So meaning that all hypotheses from Alain Prochian are quite right. You have to recognize first the glycosaminoglycans, the good one at the cell surface, to be able to then translocate into cells. And the pore formation is similar in culture cell lines and in acute brain slices preparation. And the very interesting point is that GAGs are mandatory. If you remove GAGs, so in GAG deficient cells, there is no translocation or very poor translocation. And translocation is boosted by hyperpolarization, so the former hypothesis from Paul van der is right as well, because he focused on the role of membrane potential at the beginning, and we found the same. But you have, the GAGs are needed. Hyperpolarization cannot save translocation if you don't have GAGs at the cell surface. And so the experimental conditions, so peptide sequence, concentrations, as shown also cell lines, because they have also different resting membrane potential. So the internalization capacity of the peptide is different because just of experimental conditions, but in all cases, the pore formation is very similar, at least their duration. And so, as mentioned, the kinetics are very much quicker than what has been shown for all endocytosic processes. So this is just the final images to draw how we see things today. So you have glycosaminoglycans on protein. On this and induce endocytosis, and or they can also use the interaction with CSC to mediate translocations through pore formation. So just I would like to finish with so these are people who have been involved in some parts of the story I've presented today, and thank you for your attention.
Thank you, Professor Sagan. Do we have any questions? We have time for questions. Yes.
I have a dilettantish question. If you are working with these arginine peptides, transfer peptides, CPPs, either 9 or 8, they penetrate very efficiently into cells. If they are there at high concentration, what's the interaction with native nucleic acids?
Sorry, I didn't understand all the question.
Yes,
could
if you are working with arginine-rich peptides.
Arginine-rich peptides, yes.
CPPs, R9, R8.
Yes.
They penetrate plasma membrane very efficiently. And they are in cells at very high concentration. What about the interaction with native nucleic acids, RNA, DNA?
Well, they can interact with native.
They?
Do they? I don't know.
This affinity is very high towards these things.
Yeah, I think they can. Once they're inside cells, whether they can translocate into the nucleus is another question. Penetrating has been shown for instance, by imaging, fluorescent imaging, to be able to accumulate into the nucleus and the nucleolus.
Nucleoli.
Yes, nucleoli. But for R9 or I8, I don't recall. Maybe you have the answer, Margus, or? Okay, so they do.
But how they affect cell life then?
I don't know. I'm not working on that, and I don't have any...
People have worked now 35 years with them, and they don't speak about it.
Yeah, well, how do they impact cell life? At least they are not cytotoxic. This is for sure.
How do you know?
Well, because people have tried the cytotoxicity, studied the cytotoxicity, and they didn't show, at least in cell lines, or they don't have toxicity.
But they are bound, but they have no effect.
It's always a kind of dynamics, because they cannot. It's like for GAGs. The peptide attached and detach every time. So maybe the way they interact with nucleic acids does not impact the whole life of the nucleic acids in the cells, because in the sense that they attached and detached, and probably they are not so good competitors as the natural partners.
Thank you. Do we have any more questions?
Very nice talk, thanks. When homeoproteins translocate across membranes, is it dependent on this 3-helix motif, like in Antennapedia, or there are no ideas at the moment?
Well, in fact, what we have shown in a paper is that the hypothesis is that you know that the whole homeoprotein has 3 helices, and the 3 helices are globular in terms of aspect of the homeoprotein. But as soon as so this is a modelization also process that helps us to imagine how it works, is that so you have at the upper side of the homoprotein, the gag recognition motif. And so this gag recognition motif is in the disordered domain of the homoprotein. And so it can interact very easily because it's not folded. And as soon as the protein interacts with gags, you can have unfolding, not from the helices, but from the three-dimensional protein. And then the helix 3 is free somehow, and at the close vicinity of the lipid bilayer, and then it can interact with the lipid bilayer. While when it's folded, it's not that accessible. So the gag binding promotes the unfolding of the three-dimensional conformation of the protein, and then it allows the interaction of the third helix with the lipid bilayer.
Thank you.
You said that hyperpolarization changes this whole dynamics. If I recall, the whole homodomain story started by morphogenesis of the nervous system, excitable membranes.
Yes, well.
So would you devote a thought to what happens if you have excitable membranes, you hyperpolarize? What is the resting membrane potential at which you made these statements that, it is much faster than translocation, the size doesn't matter. These are pretty
obviously
things.
Okay, a lot of questions in one. Obviously, the size doesn't matter because homeoprotein, we found in the paper, hopefully that we can publish soon, that homeoproteins, the pore properties of the homoprotein is similar than to CPP. So you think about 8 amino acids or
not 60 anymore.
Compared to the whole size of the protein. So they have the pore property are very similar. Okay, so they have, so if you think about hyperpolarization, in fact, there is 2 things. You have to have the presence of the good gag at the exterior of the cell, and you have, if you are hyperpolarized, you can think that the internalization is faster because, of course, homeoprotein has a basic PI. Okay, so once the protein is within the membrane, then it attracts more, better than in a cell that has a resting membrane potential of minus 75, for instance, as we can be found in neurons, compared to a CHO cell line that has a resting membrane potential of minus 25 millivolts, for instance. So meaning that that's a huge change. Yeah, but if you don't, so it works. It works whatever the membrane potential, but for CHO cells which have a resting membrane potential of minus 25, if you go to minus 75, it works better, meaning that the frequency of pore is higher, is deeper, because I think that but the duration is the same, meaning that once the pore is formed, because the attraction inside cells is higher, then it goes faster. And then it's the membrane closed, and then is ready for another peptides to interact with. So it means you increase somehow the efficacy of the peptide by the hyperpolarization. But this can be done only if you have the correct GAG present already at the cell surface. Because if you do not have, it's not working. I mean, this is the group of Alain Prochian who have shown, for instance, that if you take OTX2, which has a gag recognition motif of CSE, as I show in here, so in perineural nets, you have a lot of CSE. And this homeoprotein accumulates in this region of the brains, compared to, for instance, N-grade 2, which is, I mean, we here have shown that in N-grade 2, the gag recognition motif targets heparan sulfate and not CSE. And this homeoprotein does not accumulate in perineural nets, even in the presence of minus 75 millivolt resting membrane potential. So meaning that the evolution has done the things very well. So you have to have the correct partner at the exterior and the correct conditions in the interior to make the system specific somehow.
Thank you, Professor Sagan. Thank you. And now I will ask from our technical assistant that Professor James Eberwine is online, I understand. Perfect. Perfect. Unfortunately, he could not join us in person, but thanks to our technology, we have him here anyway. So Professor James Eberwine is calling us from University of Pennsylvania. And his research explores how cell-penetrating peptides can function as programmable systems to transport molecules inside cells. So the floor is yours.
Thank you. Can you hear me? Hello, can you hear me?
Yes.
Oh, great. Thank you. First off, I'd like to wish you all a good afternoon. I'm sorry that I can't be there in that beautiful room to help celebrate Ulo's 75th birthday. I especially want to give hello to Ulo on his birthday, but also to my dear friend Thomas Bartfi, who's there. And I'm so sorry that I can't be there to see and interact with both of you and the other conference attendees. I also want to thank Dr. Spool for his help in organizing this, and his help was greatly appreciated. So what I'm going to do today is tell you a little bit about how we use cell-penetrating peptides. I'm not a chemist like probably 90% of you in the room are. I like to use these reagents to try to query cell biology. And so they've helped us in a number of different ways, and they continue to help us as they improve in cell specificity and ability to get into cells. And again, I'll show you some of the things that we've been doing, some in collaboration with Ulo. So for us, that's exactly what we do. We try to use cell-penetrating peptides to manipulate cell biology. We're not so interested in therapeutic aspects of CPP, although of course I think that's very important and many of you are doing that. What we want to do is understand fundamental processes, how cells function. And CPPs help us bring our reporters into cells so that we can then assess how cells respond to various types of stimuli, their natural microenvironment, and other types of manipulations that we choose to do to the cells. Cell-penetrating peptides have been especially helpful for us since we work in neuronal systems, and neurons are especially difficult to transfect with standard transfection protocols. We've been working on various procedures for introducing RNAs and other molecules into cells, in particular photostimulation and phototransfection, having developed methods that allow us to use light. But another approach is obviously the cell-penetrating peptides, and using these in conjunction with lipid nanoparticles, which we're doing in our lab, and I think some of you are doing, using it in addition to light activation, all make for high efficiency introduction of associated compounds into the cells. I do want to highlight that Ulo has been very involved in all aspects of this, as you're all aware. You're all there to celebrate his contributions. But one of the things that I've always been impressed with was Ulo's ability to sort of see the future, and his ability. And when he went in and synthesized the first synthetic CPP, that really was a conceptual shift. It shifted CPPs from just naturally occurring domains into products that can be rationally designed. And I dare say that probably most of you spent large portions of your career trying to use these methods that Ulo pioneered to develop CPPs that meet your experimental needs. One of the things I want to highlight, I don't know if anyone else has done this, but Thomas Bartfai, with whom Ulo worked many years ago, was prescient in every aspect of science, but in particular with respect to today's talk, in having one of the first peptide synthesizers and allowing Ulo to use this for his initial synthetic efforts. And so good science and new science is always driven by technology and available technology. So it was nice that Ulo had this available to him. So I'm going to show you a few different biological uses of CPPs. This first story is one that was inspired by Ulo, but we did not collaborate with Ulo. It was inspired by my many years of working with Ulo in the past, and we worked with Ivan Mahovsky here at Penn. And what we wanted to do is try to capture RNA in live cells in real time. What most people do these days, I'm interested in single cell analysis, single cell biology, and subcellular biology. And we want to query the molecules within individual cells and subcellular organelles. What we'd really like to be able to do is not kill the cell to isolate the RNA or the molecules we're interested in. We'd like to leave them intact and live and still query the molecules while they are alive, or be able to know with a mark at what point we initially looked at the cell after we isolate the material. And so several years ago, we developed a procedure called TIVA, which is transcription in vivo analysis. And I want to emphasize each of these vignettes is going to have in vivo in yellow, because we want to work in live cells as much as we can. So this is the in vivo capture of RNA in live single cells in real time. And so we designed this compound that you can see. I don't know if you can see my arrow, but it's here on the right-hand side, where we have a cell-penetrating peptide that will carry this into a cell. We have two regions of nucleic acids that anneal with photocleavable moieties within these these short stretches of nucleic acids. And we also have a biotin on the end here, and you'll see how we use this momentarily. The idea is that this will go into a cell. Then when we hit it with light, this will break and this will break, allowing the poly-U to bind to the endogenous poly-A RNA that's in the cell. So for those of you who are not molecular biologists, probably a good 95% of the RNA in the cell has a poly-A tail. All the mRNAs generally have poly-A tails, so we're querying the mRNA fraction of the cellular RNA. So the way in which this works is the TIVA tag is made. It goes into the cell by virtue of the CPP. CPP comes off by the natural reducing environment. You have a FRET signal where it's connected, and then when you hit it with light, that FRET signal disappears. And so you can see activation of the compound directly in the cell after the compound is in and after you then activate it. This is an illustration. The CPP, the compound's in the cell. You hit it with light, and now you've lost the FRET signal. So you know that the compound has been activated. Once activated, it will bind to the poly-A plus RNA that's present in the cell. And then you can use the biotin moiety that is on the capture sequence to pull this out of the cell when you harvest the cell. So this is a way of looking at RNA at a particular time of stimulation or inquiry, but then you can isolate it afterwards. We've done a lot to assess the robustness of the technique and what you can see here is some of the data. You can look the paper up if you're curious about how it works. One of the important things is in the lower right-hand panel, you can see sequencing reads that map to a particular RNA that we found in just straight RNA, in RNA that was isolated with the TIVA approach. And you can see that we're getting the same reads, same exons identified, and this shows that the technique is working nicely. One of the things that we found, again, the data is in the original paper, is that when you do analysis of RNA in dispersed hippocampal pyramidal cells, this is all from the mouse in this case, we find about 12,000 RNAs that we can detect transcriptionally from a single cell. When you look at a cell that's in intact slice, a hippocampal slice that's alive, and do the TIVA approach on those individual cells, you find about 9,000 mRNAs. So you find less RNAs on a cell that's in synaptic connectivity and neuronal connectivity with other cells. So the other thing is when you look at TIVA in tissue, the lower graph on the left-hand side, you find much more variability in the expression of RNAs, as one might expect from individual cells, where the expression levels could be quite varied. When you look at mixed primary cultures or whole tissue, you have an averaging of all the RNAs, and those that are present at lower abundance, which would be towards the right-hand side of the red graph, would be missed by the technique that one would normally look at. So TIVA allows you to identify variability within the cell and the large number of RNAs that are present within the cell. And one of the important points of this is the endogenous microenvironment, or the connectome, acts as an inhibitor of overall gene expression and facilitates cell-to-cell transcriptome variability. So basically, when you have a cell in a tissue slice connected to the other cells, there's less gene expression than you find in a cell that's dispersed. This bears on a lot of the work that many of us do on cells in culture, and just sort of keeping this in mind as an important component of how we think about the data that's generated in dispersed cell work versus intact slice work. We use the TIVA, and these experiments were again prompted, the cardiac and next couple of slides, inspired by Thomas's urging of me to look at other tissues. And so here we've looked at mouse cardiac tissue. So this is sliced through the mouse heart, and we can see activation, take up of the TIVA compound and activation within the cardiac cells. And you can do it in pancreatic islet cells as well and assess the transcriptome in those cells. What I've been keenly interested in for many years is RNA that is translocated to different regions of the cell, in particular in neurons, to dendrites and synapses. It's at those sites where learning and memory occurs, much of synaptic plasticity occurs, and every neurodegenerative disease has some type of problem, dysfunction associated with neuronal synapses. So this is a site of neurodegenerative disease, symptomology, and other types of illnesses. So we want to understand what RNAs are there and how they might be locally translated. So we wanted to do this in live cells. In the past, we've actually isolated dendrites and looked at the RNA complement in the subcellular regions. But we want to look in live cells if possible. So here we put the TIVA tag into the cells via the CPP, and then we photoactivate the TIVA compound only in the dendrites, not in the cell soma and subregions of the dendrite, as illustrated here. This is an example of some of the work. So here you can see TIVA in the whole tissue, and we can activate an individual cell by shining the laser light on this. And then when we use 2-photon activation to be as precise as we can see individual dendrites here from the TIVA tag. When we hit it with light of the appropriate wavelength, we activate the compound. And you can see it is green, and then here, this is an individual dendrite, and then down here, this is an individual dendritic spine on one of these dendrites. So the compound is present throughout the cellular cytoplasm, even though you can see a large amount of it in the nucleus, and it can be activated in these subcellular regions. When we do that, actually, this is just another example that I just like to show since I think it's a pretty slide. This is the intact, a live slice through the cerebellum. In this case, it's human cerebellum that we harvested at the time of neurosurgery, was harvested at the time of neurosurgery and brought to our lab, and we can keep these humus specimens alive in our lab, and both in dispersed cell culture and slices. And so you can see the TIVA compound in the cells of the cerebellum, and then you can see, again, I hope you can see my arrow, but there's a white arrow pointing to this dendrite. Here, this is unactivated, and here it's activated, so it becomes blue in this case. And you can see that this works nicely for these cells in the life slice. One of the important things about this for neuroscience is cells generally function in context of the cells in which they connect with, and being able to assess cellular biology in cells in their environment where these connections are intact is a critical component of understanding how cells actually do function, particularly in vivo. I won't bore you with all the detail, but when you look at the transcriptomes, I believe you can actually categorize neuronal type and disease state based upon the dendritic transcriptome itself, not just the whole cell transcriptome. I showed this one already. And then in this example of human cerebellum, you can see the individual areas that are green on the far right. Those are the areas where we did the TIVA analysis. We pulled the RNAs out, we sequenced them, and you can find in this region of the dendrite, in this particular dendrite, there were 533 RNAs, this one 398. The fourth one down didn't work. And so we can identify these RNAs in these subcellular sites in human tissue using the TIVA tag, which is facilitated and made possible by virtue of the CPP, getting this into the cell. We modified this to be able to look at not just the poly A plus RNA, but all the RNA in the cell. And so we engineered a TIVA compound through ruthenium nucleation, where we have a degenerate sequence, not a poly T sequence, which will bind to poly A. This will bind to essentially any sequence. So this will carry this into the cell. We can use photoactivation to liberate this. In this case, you can see that we only have a single strand. It's not double-stranded in the TIVA compound. And when we put this in the cell, the idea is that the degenerate sequence would interact with essentially any RNA sequence. And when we do this and look at gene expression for TIVA tote, which is all the RNAs versus the poly A plus RNA in the cell, we find a nice correlation between the two. So the TIVA tote is seeing many of the same RNAs that the poly A plus is seeing, and in addition, other ones that are not described here. Well, actually, some of them are like the microRNAs. We're seeing 124 different microRNAs with TIVA tote that we do not see with the TIVA poly A plus. I'm going to switch now to another application of CPPs, and this is one that we did in collaboration with ULO and several years ago. And this is an effort to try to look at RNA binding proteins associated with particular RNAs. All RNAs in the cell are covered with RNA binding proteins. And so we want to be able to identify the endogenous RNA binding proteins that bind to any particular RNA. And so we call this PAIR, PNA-assisted interrogation of RNA binding proteins. So in this case, we have a compound that has, I can't see it on my screen, but down here at the bottom, you can see a CPP connected to a PNA that has a BPA photoactivatable group on it. And so the CPP carries this PNA, antisense PNA, into the cell. In this case, it'll bind to the ONC mRNA. And once it binds to the ONC mRNA, we can hit the cell with light that will activate the PPA. It'll cross-link to any proteins that are near that site in the RNA, and we can then identify the RNA binding proteins that are near that site. And so when you do this in cells, you can see over on the right-hand side the position of these particular CPP-directed PNAs. And you can see that nucleolin is present on all three sites within the RNA. RNA PK seems to be restricted only to the 3' end, in particular this PNA. the sequence. And so there's commonality, but also specificity to these interactions, which has allowed us to really understand these interactions between RNA binding proteins and RNAs much better than the standard techniques that people use in terms of immunoprecipitation and other approaches that do not have the single cell resolution that this does. Now, finally, the last part, this is work that has not been published. This is work that Ulo and I worked on for many years. Unfortunately, we were not able to bring it to completion. You'll see it at the very end. But it's a great idea, and it's something that it would be wonderful to get back to, even though it's 25 years later. Ideally, we'd like to be able to look at transcription in real time as it's occurring. And so TIVA allows you to capture the RNA from live cells, but it doesn't look at transcription as it's occurring. Transcriptional dynamics would be, at the level of single cell, would be the ultimate goal of this type of approach. So we'd like to be able to look at real-time detection of RNA transcription while identifying the RNAs in live cells. So to do this, Ulo and I developed the blinker technology, and I really want to acknowledge Ulo's integral involvement in this. He helped design the complex peptide that we worked with. It was difficult to make in that time, and his creativity was very important in making this work. You can see it blinking here. That's what I'm trying to show with this. So the idea is we want to modify endogenous RNA polymerase so that as the RNA is coming off of the RNA polymerase as it's transcribing DNA, you detect the RNA as it's coming off the RNA polymerase. We know a lot about RNA polymerases from the 3D structures that have been categorized, developed by a number of different people. The imaging is driving this particular set of experiments using the structure to identify areas that we want to try to then query. And the idea is, that there's an RNA exit pore on RNA polymerase. And as the RNA is coming out of this, we want to be able to query it. And to do this, we've developed a peptide called blinker that has been engineered to bind to a region of the RNA polymerase. When it binds there, it has a fretted PNA. And when RNA comes out of a particular sequence, the fret signal is lost because the PNA anneals to that sequence. So you can imagine as the RNA is exiting the pore, the peptide is querying the sequence, and when it finds the appropriate sequence, it blinks. And so you can see it sort of depicted here. So the peptide is bound to the RNA polymerase as this moves down the DNA, and the RNA is coming off. The blinker peptide queries it. This allows us to identify RNA sequences because, in this case, the PNA in the blinker peptide is GTG. That will query CACs, meaning that we'll anneal to CACs. So as the RNA polymerase is going down the DNA, and the RNA is being made, as this RNA is coming out of the RNA exit pore, if there's a CAC, the GTG will bind and get a fret signal, a blink. You can see the CACs are at different positions within this RNA and different positions in this RNA. So the positioning of these CACs in the RNA is diagnostic of the identity of the RNA. So it's essentially the sequence of individual RNA. So if you can look at those sequences, you can and query them, you can identify the RNA just based upon this trinucleotide and the position within the RNA. So of course, the position of the RNA in our blinker approach translates into time between blinks. So the time between blinking here and here is different than here and here, and this would be different for this RNA. So the time for blinks is the signal for sequence identity. This is a little movie. I had a postdoc who really liked making these little movies. So you can see the RNA polymerase with the blinker peptide on it going down the DNA. It finds the sequence of interest, it blinks, and you can detect the signal. And so I'll let it go one more and then we'll stop it. You get another blink. So the distance between these sequences is reflected in time of blink, difference between the blinks. So the technological basis of the blinker is we need a transcription detector, the blinker peptide. That was Ulo's effort on this project. We need to modify the RNA polymerase so it can bind to the blinker peptide, and we wanted to make blinker arrays. That was my job. We needed to develop an imaging methodology for visualization of blinker. That was again my job. And then Junyoung Kim was involved in the data analysis. So we wanted to test this. So rather than going to the endogenous RNA polymerase to begin with, we tested it with T7 RNA polymerase. So I made several different T7 RNA polymerases where the capture sequence for the blinker peptide was engineered to have different turns in the capture sequence. So it would have different ability to bind to the blinker peptide. I wasn't sure which structural conformation of that capture region on the RNA polymerase would be best for the blinker sequence to bind. So we had to engineer several of these. And one goal of this experiment, and I'm going to show you some of the data on this, is we'd like to be able to take cDNA libraries, even genomic DNA libraries, cut them and put them onto blinker-ready arrays. So blinker-ready arrays in this case would be arrays that have double-stranded DNAs that have a T7 promoter on them. You ligate your cDNA library, or I'm sorry, you cut it with restriction enzyme, you ligate it onto these oligos that are immobilized on the array. And now that means that you can add T7 RNA polymerase. It'll go traverse the DNA and make RNA, and you should be able to use the T7 blinker approach to query that RNA. Because of the speed, well, that's not sure. Yeah, here it is. I'm sorry. So these are some of our test efforts on this, where we take a DNA with the T7 promoter, put it onto the array, and use our blinker peptide to query this with T7 RNA polymerase to put onto the array. And given the speed at which the RNA polymerase moves down DNA, which is about 80 bases per second, you can get full transcriptome analysis within 2 minutes. In fact, it should be faster than that, but we did 2 minutes for our analyses. We did not do full transcriptome analysis, we just did the testing, but that was the goal, and you'll see that it's enabled by this. So rather than going on these DNA sequencers that take overnight to sequence the libraries that are on them, this would facilitate very rapid analysis at much lower cost than the standard DNA sequencing abilities. So we tested this on when we used 2 different DNA templates and used the blinker analysis. We were able to get 10 correct annotations of the sequence out of 12. So we missed 2 of them, but this was using blinker to blindly sequence and detect the DNA templates. When we went up to 9 templates, we only had 33% accuracy. That's still not horrible, but it's certainly not at the level that we would have liked. I think that was likely because of bad template choice on my part, and also limited signal to noise detection. So this is one of the major issues. We did this work 25 years ago. This was before AI analysis. And trying to identify the signal within the noise. This is a very small signal, as you might imagine, single molecule detection. It was very difficult. And I reached out to a number of different people, including people at IBM and others, to try to help with pattern recognition, but they really weren't interested in biology at that point. And so this was really one of the major limitations. One of the limitations was making the peptide, which was difficult. The other limitation was the analysis of the data, which was difficult. I think this would be so much better now with the current AI pattern recognition algorithms that are out there. And so it would be nice to be able to try that. We'd like to be able to move this in vivo, not just sequence libraries on arrays. So we need to be able to put this into cells. So we did in vivo blinker pilot experiments where we put T7 RNA polymerase into the cells, the modified T7 RNA polymerase, and put our blinker peptide into the cell. Let me show you this. The compound on the left is the compound that Ulo and the people in his lab made. You can see how quite complex it was, particularly for 25 years ago. But this did go into cells. We could identify the fluorescent compounds within the cell, and we could stimulate blinker, we could activate it. But we did very little transcriptional analysis. Unfortunately, people in Ulo's lab that were involved in the blinker peptide production went on to more permanent jobs, and the AI analysis and informational analysis from my lab was really not nearly what it should have been to be able to get through this. Nonetheless, the idea is there. The idea is still valid. Ulu and I and other members of our team won an NIH single cell prize for this technology in a competition to try to do in vivo analysis. But unfortunately, we never brought it to the point of publication, even though it was very significant then. And Ulu, I think, this still remains significant. Finally, I'd like to thank Ulo for the scientific inspiration and the several collaborations we've had. Cell-penetrating peptides are amazing moieties. They've allowed me and others to look at cell biology in real time. They've helped people with therapeutics. I wish I could have heard the other talks at the symposia, because I would have learned a lot about the newer applications of cell-penetrating peptides. And cell-penetrating peptides do everything, including, in this case, carrying Ulu's birthday cake into cells so that they can be attain sustenance from the creativity that Ulu has brought to all his collaborations. So thank you for your attention. Ulu, happy birthday. Thomas, I wish I could be there to see you. And my greetings to everyone in the audience. Thank you for your attention.
Thank you, Professor Eberwein. Do we have some questions for him? I guess you were very clear. Thank you very much.
Thank you.
Thank you. So we will move to our next speaker, who is Professor Tamas Partfai. He's Professor Emeritus at Stockholm University and Professor Emeritus at University of Pennsylvania. And as we have heard, he has played an important role in several people's lives presented here at the moment. He is distinguished neuroscientist and pharmacologist. And today, he will discuss the design of chimeric peptide antagonists and cell-penetrating peptides.
You see, I have resources. You kept one, but they gave me one. But this is not as good as yours, or the talk will not be as good. I will say because of it. Thank you very much for inviting me. I am not an expert on cell-penetrating peptides. I was trained as a mathematician, and then a nuclear physicist. And then I got a job in Sweden to do the dishwashing in a biochemistry lab. And by delivering the not so clean glass, I discovered that the experiments which people did, very nice people, very clever people, were poorly designed and poorly evaluated from the point of a mathematician. So I didn't find any other way than politely explaining to the guy who was running the lab. And he said, would you mind writing a thesis on this? So I did. I submitted it, and somebody gave it back to me, and he said, you are not from the north. You are not Scandinavian. You are born in Hungary. One can hear it from a mile. You are not 25 years old. How many things do you want to do? And I said, well, I don't want to sit in jail again. So that was kind of my goal. My other goal was to stick with what Albert St. George said to me. And the man who trained all the electrophysiologists, you have used the work of Kufler and Eccles. They both said that scientists, unlike poets, even if they come from very small nations, they belong to the whole world. With poets, it's a tragedy. if he comes from a small language, if he leaves it to write in another one. But for scientists, it's a must. It doesn't mean that they left. They never leave. We are here. He is here. He is here. Markus is here. Many are here. You are back from Maastricht. So what you will hear is basically me thinking about if I am going to make one more trip. It was agreed on after some strokes and so recently that I don't travel anymore. But I did come because Ulo is one of the very few close friends I have. It's very hard to keep truly close friends. The people who have many, many friends, they don't know what a friend means. They mix it up with acquaintances. I don't. This is why I am here. What I appreciated is that Ulo, who ended up in the lab on recommendation of Jaak Järv and others, for which I am grateful, certainly worked with me 39 years. That's like 6 times the average marriage in the European Union. And we didn't have a fight, which can mean only 2 things. He's extremely diplomatic, because I am not, or he's extremely smart. I think the latter is more likely. Also, I don't want to get involved in the work of people just because they made a discovery in my lab. I have been asked by Tiit last time, when we spoke about Victor Muth, who discovered more neuropeptides than the whole industry together, but he spent all his life in Sweden. It was nice to have a symposium in his honor here in Estonia, because he was born here. And we spoke about it, that I had 19 nationalities in the lab, and altogether over 250 people, postdocs and PhDs, between Yale, Stanford, Scripps. So Jimmy Berwine I met when he got a full stipend to Yale and sent him to Stanford. I insisted that he goes to the best place right now I could find, so I had to call the head of the National Cancer Institute. He said to me, "Don't tell me that he is the best you ever had. Everybody always says that. Say something different." I think you had a good time there, scientifically and otherwise. And the same is true for the others. There had been very, very few fights. When I had to build a new lab and a new institute for neurology to succeed Floyd Bloom, who went to become the chairman of Science magazine and the founder of the National Institute for Drug Addiction and Alcohol Addiction, because he believed as a psychiatrist that addiction is a disease, it's not a weak character. It was not so trivial in a very much more Republican America than today. It was not trivial. Anyhow, he specifically came to Basel and recruited me with Jerry Edelman. He said, "I want you to take my lab." I said, "Why?" He said, "Because you are broad." And in most places, being broad is not a great compliment. Being broad means you are not that great in anything, but you have some kind of journalistic know-how of everything. I hope it's not the case. What I appreciate most in Ulo, besides our personal friendship, is that when he finds something which is an experimental result, which most of us would think depends on some mismatch of experimental conditions, real mistake, then he doesn't give up. So when the peptide, which was supposed to bind on the outside of a cell, because it was designed as a receptor ligand, and that receptor was facing outside. And the person just weeks before he came was Al Gilman and Martin Rodwell in my lab, and they worked on the G proteins. Both of them were sitting in the lab. He was still somebody on some boat on the way to Sweden. He had a very nice light gray suit in which he arrived. I still suspect it was from GUM. And they told me, you know, none of the ligands of neuropeptide receptors will ever spontaneously go into a cell. You will have to go so far as ferritin, and so if you want to get something into a cell. And this is why we work on second messengers, and then I worked with Paul Gringard, who worked on what a second messenger can do when it activates a kinase. But I didn't ask Ulo to work on that, not at all. I will tell you about this machine. I somehow, if any, I have an attraction and a talent to original people who are smarter than me. And there are many of them. So this is Ulo at the bench. He had a white rock 39 years ago. So now I have to explain to you that I was terribly attracted to discuss with Bruce Merrifield at Rockefeller. I ended up at Rockefeller because I was an advisor to Nelson Rockefeller, who was governor of New York and who lost to an extreme right-wing candidate the Republican nomination. But Rockefeller had owned and founded 6 universities, including Chicago University, which is fairly good, Rockefeller University, which is fairly good. And I was working at Rockefeller, and one day I was called in. The governor wants to talk to you, wants to have advice. Advice from me? Do we keep on malaria research? And I said, as far as I understand, it's not solved. And it's still the major place for malaria research. After that, I became an advisor to David Rockefeller, his son, and their grandsons. The grandsons are now living in Australia and New Zealand because I told them, together with many other people, if your name is Rockefeller, you can't have a normal life in the US. You will be different. But David, he became a senator for West Virginia, which was the poorest state. And I saw that the only way to lift public health if you start to use the place for clinical trials. Because before somebody is enrolled into a clinical trial, you measure blood pressure, blood sugar, trivial things which are easy to treat if you know about them. The advisors of the good senator called me and said, if you once again come up with such an idea, we will shoot you. Because who will pay for those tests first? If we lose the elections. Nevertheless, I remained at Rockefeller, and there was a man who had a very simple idea. Any one of you who has ever been asked to take care of knitting knows that even though it's a big roll, it's only one wrong thread which you knit with. And that's the single idea of Bruce Merrifield. He said, It doesn't matter that the folded protein is globular, elongated. It's a single polymer, which is a linear polymer. What it means is that you can fasten one end and then build it from there. Until that point, every one of us synthesized peptides in solution. And we had to come up with 55 protective groups for every amino acids. The simple explanation he had, it doesn't matter, it's a ball. Actually, this has one end. If you fasten that end, then you can build it. And then later it will take a confirmation. Bruce Murrayfield was a genius. Unfortunately, very early died in cancer. We all loved him because he was one of the most modest people there. The person who ran the lab for him was Jimmy Tam. And since I believed in sending to the best places the best people, I had a guy who worked on a Victor Muth peptide, the first one which Victor came. He came Friday afternoon, Victor Muth, said, we isolated the first one not from the intestine, but from the brain, and we called it neuropeptide Y. Because the only amino acid we discovered is tyrosine. It starts with the tyrosine, the Y stands for tyrosine. On Monday afternoon, he already came back and asked, did you synthesize it? We haven't. Eventually, I had to rely on Ylö, who was organized, original, and didn't complain. The moment on, he took over the machine. We could do it. So Anders Sundén got a PhD with me after having been to this lab, and he did a little modification. The modification was that to make solid phase peptide synthesis where the C-terminal is amidated is easier and cheaper than when it is a carboxyl. And that was the same idea as Victor Muth had. Victor Muth only wanted to isolate peptides where the C-terminal was amidated. He said those are unlikely to be fragments of proteolysis. You see how my word comes together? So I sent Anders there. Anders, who would never go to America for any million ideological reasons, loved Rockefeller. He loved it. You know that. So he came back, and the problem was that nobody, absolutely nobody wanted to build a machine to do this now. And so we got together and we built a machine with the guy whom we knew how to build machines. His name is Lee Hood. He built the first sequencing machine, and eventually a small company was formed by Sidney Brenner, Lee Hood, and myself. The only reason we formed the company was because nobody would talk to us when our card only said Dr. So-and-so, even though Dr. Sidney Brenner had already a Nobel Prize and 3 Laskers. That was not good enough. But when it said Dr. Sidney Brenner, Chief Scientific Officer of Company X had 0 capital, but it was a company. All of a sudden, everybody wanted to give us money. We built the synthesis machine. We needed somebody to buy the number 2 machine. The number 1 machine remained at Caltech, where it was built from the money which David Baltimore gave, who was then president. And I believe in sending people to, I sent several people to David, 4. All this cDNA which Jim was talking about, the transcriptome, which he teaches, he taught 24 years, Jim Eberwein, the previous speaker, at James Watson's seminariums area. all this is based on transcriptomics, but that was impossible to do. Even though Sidney Brenner predicted that there is mRNA, nobody ever could isolate one. It was too short-lived. But the reverse transcriptase, which David Baltimore discovered in retroviruses, made possible that you could turn into very solid, very long-lasting DNA, your mRNA. So my word, in every way, is together. So I needed somebody who would pay for the number 2 machine. It was very expensive. It was not made in thousands, still not made. And so I went to a man named Bertil, the best friend of Victor Muth, by the way. They were together. He discovered heparin, fairly important, I should say. And by that time, I asked him if he would get me a machine. And he just founded a company called Scandigen for the same reason. Nobody wanted to do gene-based company in any country, not in America, not in Sweden. So Kobe, where he was the boss, didn't want to be known to be involved with this very new technique, and therefore they founded a separate company, which is a private company called Scandigen. It was actually run by a man who had a publishing house and loved poetry, Thomas Fisher. Nowadays he owns, I think, Mauritius Airways, but I don't know. Don't ask me why. And so he bought the machine, which we knew from here, through him. And then they asked me, what do you want to do? And I said, I only want to make vaccines, because clean water, food, and vaccines are the basis of life. And at that time, there was 5 million children dying in Africa in whooping cough a year. Sweden had regular epidemics of whooping cough. So I wanted to make a whooping cough vaccine. Turned out nobody has ever sequenced the toxin. So we sequenced the toxin with Lee Hood's other machine. You know what happened? It turned out that the vaccine was easy to make because forgive me, because the pertussis toxin is a very special protein. I will show you in a second. So again, nobody wanted to give us money, so we formed a company called Trion. I didn't know that I was in it. Nobody asked me. So Bertie gave the name that we three had a company called Trion. Trion all of a sudden owned the only synthesis machine and the only sequencing machine. So once we were at there, we did pharmacophore mapping, and Tit came in, and he did the first really good pharmacophore mapping of galanin and showed that tryptophan to tyrosine 9s are key. And he also showed, as he showed it to you, that the first 13 amino acids are important. And that was the basis of the chimeric peptides. But the peptides themselves, the tons of them compared to what existed. There were other people who made peptides with bags or so. We were not unique, but we had a mechanical advantage because Lee Hood is a mechanical genius. And so from that on, Ülo made galantid, galanin and terminus, substance PC terminus, And I didn't bother him after that. When I discovered that he puts my name on a paper, I asked him, what did I do on this paper? He said, first of all, you will read it now and help me to correct it. And secondly, you can recommend good experiments. That was about what I did. True or not? I did not get involved in what he did. They decided that alanine screen would be the best. Today, there are more than 6,000 papers using alanine screen. So if you give good tools to good people and then you send them to good places, something good may come out. That's my conclusion. So what happened when we have synthesized, when we have, so Bertil was CEO of Kabi. Kabi formed Kabi Jane, but then somebody on the board said it's too modern. We are selling heparin. That's our major product. The second major product was growth hormone for every company who was selling blood products. And the growth hormone was purified from dead people's pituitary. And we were always worried that somebody has a virus which we don't know of, and therefore we don't test. Sure enough, HIV came, and therefore did we want to be able to make growth hormone something else. And so Matthias Ullén and I went to Genentech, where two of my old classmates were just trying to make the first growth hormone. But we were so uninterested in the business that we only took the rights for the Scandinavian countries. We didn't take the rights for the US or Japan. I mean, it's unbelievable, but it was so. You can check it. So you know why Genentech became valuable, but because I bought it for Roche with Charles Weissman, I had to learn how valuable it became. But we were so uninterested in the value of it. We were interested in the process. And so when we came to the pertussis toxin and we sequenced it, it was a complete surprise. In 158 amino acids, there is not a free amino group, not one. There is not a lysine in the whole sequence. Every 20th amino acid should be one. It isn't. And the N-terminus, as for most bacterial peptides, is acylated. So there was not a single amino group, and therefore the way how you toxoidate with formaldehyde couldn't work. So every batch of pertussis vaccine in the world was different, and therefore unreliable. And people missed out not only on having protection against whooping cough, which caused 5 million deaths in Africa per year, but also they missed out on tetanus and diphtheria because it was given as a triple vaccine. The children didn't have diphtheria, tetanus, or whooping cough. So these are the two papers. Here I put my name, and you'll see he worked on it a lot. And as you see, there are people from the Balticum. And he was as close friend of mine as Ulo is, but he's gone now. And he is also gone. They were both 5 years younger than me. So what happened? That I am interested in how does an interesting project comes about. And this is the key slide I came up for. So you see, these are unrelated projects which Ulo made a story of, not because that was the first peptide for intracellular transfer. The homodomain of Prochyons did that. The TAT sequence of HIV did that. But this was the first one completely synthetic, and the first one where we started to make very large libraries, very large pi. That's nothing is large enough for AI. On the other hand, AI is the most disgusting feature of intellectual life because it doesn't generate any new data. The Chemistry Nobel Prize was given for all the data used in crystallography, which took us 150 years to collect. But they did not pay for one single crystal. I am late. I will finish very fast. So out of these projects we made many peptides. We made a safe vaccine. This we went to Soros because he had a project to help Africa. And we said, how about, since women do most of the work in Africa then, this was before the Chinese started to buy up Africa, if they don't have children dying or being sick with whooping cough. And he gave money. The pharmacophores form mapped not only for galanin, but for many, many other peptides. Ülo has created a whole school of chimeric peptide-based cell-penetrating peptides, but that was not what was for me interesting. What for me was interesting that people would call us, whom I never before had anything to offer to. They would call me because they saw that we worked on a neuropeptide, and they said, does it have any effect on feeding or on insulin secretion? I said, yes. They said, can I have it, and can I have an antagonist? And I said, you can have the peptide, you can have a ton of it. We know it now, we make it with a machine. Unfortunately, substance P took 31 years to the sequence and another 11 years to the first antagonist. So compared to that, the first galanin antagonist is an incredible world record. And it was not one, but many. You had to inject it ICV, or you had to inject it intrathecally if you studied pain, but they worked in vivo, and he made them. So these are just the abstract. This was a key paper of teeth. This is the one which I sent to the man at the National Cancer Institute who was the doc, and he hired him. And if you open a commercial publication, bench catalog, then you can see that they already call it the first generation. This is, I think, 1 month old. And they also point out that the different ones distinguish between the subtypes of the receptor, which was a complete extra, a plus. And this is their text. And so this is what happened. I don't know why he chose mastoparan. He never told me. I never dared to ask. I don't know why he called it transportan. I had no point of view on that. I was very interested in mastoparan because every month or so I would be sitting in the same Nobel Committee talking to the man who discovered histamine release in allergic shock from the mast cells. And every time he would stand up and he said, I am against that we give a Nobel Prize for prostaglandins because there is no known illness which is caused by the lack of prostaglandin. This was Börje Upnäs, chairman of pharmacology at the Karolinska. So when I was asked, why don't you apply for his job, he will retire. I said, because I don't apply for chairs. I don't want to have a paper which will tell me what I already know, that I was not born in the Nordic countries. I'm too young, and I was not trained in anything you appreciate. So I don't apply. So then I got a letter that said, you are appointed by the Prime Minister to succeed Bengt Samuelsson on the Berzelius chair. He happens to be also director. He was not director. Hans Wichser was director, but, you know, as Prime Minister, you can't know everything. But anyhow, I went there. Bengt Samuelsson indeed discovered with Sunebergström, prostaglandin, prostacyclin, leukotrienes, and he was a wonderful guy to succeed. He said, from now on, I understand you are in Basel. When you come, I will tell you what I am working on. Maybe you have an advice for me. Every time I came, he came in, he was the first. I said, Professor Samuelsson, thank you for coming first. If you work on leukotrienes, I don't think I have an advice. He said, it's not true, because you are working on inflammatory peptides. And as far as I figured out, what you discovered is the one which everybody was chasing as the endogenous pyrogen. But we call it interleukin-1. And we just sequenced it in the next building, Hans-Jörn, and it's the same protein. It was known conceptually for 120 years. Discovered by an artillery doctor looking at fever after an artillery injury. And he called it endogenous pyrogen. So I continued to call it that, but it's interleukin-1. Today there are a few interleukin-1s. And so, all I can say is that this, from Marcus, from Ulo, Matthias I couldn't get hold of, tells you the story. And the beauty for me was that he set out to make a galanin receptor ligand, which was not supposed to do anything, but bind to the receptor, stay outside, either induce an agonist, an antagonist-like action, or nothing at all. But he went after when the peptide was gone. And it was not huge quantities, and they had to label it, and they had to do all kinds of things. And so, did anybody else get interested? So I looked it up. There is, in 26 February, there were 6,543. So what it means is that this is the number of people who quoted it. How many others who read it and didn't quote it, we don't know. But usually the number is 3. So it's roughly 20,000 people who read it. And every third one takes the trouble to actually put it into the references. And so CPPs, whether they come from homodomain, TAT, or chimeric peptide antagonists, became an interesting intellectual and hopefully useful exercise. Useful it will be for me when you make the first therapeutic agent. I am completely sold on therapeutic agents, and these are my selection, Ulo. This is a private letter to you. These are my favorite papers of yours. Thank you very much, all of you.
probably forgot that you one day came in to a department and started after elevator, coming from elevator, going to a room, started to shout on a corridor, Mastoparan, 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 like 10 times that when you were in your room and closed the door. And that's it.
Because the histamine people were using Mastoparan to induce histamine release. There is no known species which has mast cells and makes histamine and does not reduce it if you inject.
Exactly, but you forgot that it was you who brought Mastoparan to a department.
I had no idea that you would listen to me.
And the second point is that I looked it after, and then I understood that Mastoparan interacts with plasma membrane. No, it interacts with plasma membrane. And I connected it to galanin because I wanted to have one end on the receptor and another end on a plasma membrane, and to see what happens. That was...
I had no deep thought. I cannot claim I was putting you on a track. I was just telling you that I was fed up with hearing so much about histamine.
Do we have another question? No. Okay, thank you very much, Professor Bartai. And now with the final speaker, we have reached actually the beginnings, because Professor Jaak Järv was the supervisor of Ülo Langel. And Professor Jaak Järv is an academician, and he's Professor Emeritus at the University of Tartu. And I'm afraid that we will see hardcore chemistry now. Thank you.
So, thank you very much. I thank the organizers for letting me in. My topic is oriented to chemistry, so I'm sorry for this. And I start with a very basic idea that all pharmacologists are dreaming about finding a molecule which interacts specifically with some target and makes something in cell. And of course, peptides are interacting with a lot of targets, and this means that peptides should be fantastic drugs. Actually, the situation is not so simple because, of course, we know different drugs. Insulin is the most important case here, but there are problems, and some problems are connected with stability, because peptide structure is very stable, but there are special mechanisms in cell just to destroy peptides. This is peptide hydrolysis through some proteases or esterases. And now people started to think about making peptides more stable, and the idea of peptidomimetics was just born from this way of thinking. And the attractive way of making stable peptide is to replace the carbon atom, alpha carbon atom, to nitrogen, because nitrogen is very similar to carbon, and we can get structures where the backbone is almost the same, and sequence of these side chains is not affected. We call these things, these compounds, these molecules, as peptides, and I asked artificial intelligence where this idea of naming other peptides as other peptides is coming, and it comes from 1963, but if I now recall my memory, I have seen the structures like this other peptide structure even before in some general publications or some textbooks from 1950s. But then these were not other peptides, they were semicarbazates. That's a difference, and therefore maybe artificial intelligence was not too intelligent to discover this. So, then other peptides are stable. This we know very well, because in 1974 there was a publication where specific and very efficient substrate of alpha-chymotrypsin was modified, and its other analog was prepared, and this other analog was not substrate at all. They used it as an inhibitor to make a specific affinity column for purification of chymotrypsin. This was the period I was already working with chymotrypsin kinetics. So this is a paper I remember from my early student years. So we know if we put in other group, we stabilize the peptide. And now it's very easy to make peptidomimetics. And there are many papers and many textbooks. This textbook about peptidomimetics in organic and medicinal chemistry, it's a perfect book. And on page 32, there is a chapter about other peptides. And you can read, other peptides can be synthesized very easily from substituted hydrazines or hydrozides. So, can be synthesized very easily and from these compounds. Hydrazines are well-known compounds, and even we had some experience in our department in Tartu with hydrazine chemistry. And now, as they are synthesized very easily, we were prepared to make a huge project with, to generate a huge number of other peptides, with making other scan, for example, and to generate, of course, a huge number of papers. We started with getting precursors, because other peptides cannot be prepared from amino acid-like compounds, because amino acids are stable, the other analogues are not stable. Therefore, we need hydrazine derivatives. Here must be also some protecting groups, but I do not go into details. And in reaction with quite a nasty compound, we can generate carbonyl derivatives which are precursors of other amino acid. So amino acid coupling can be made with trifoscan. Trifoscan is not so dangerous as foscan itself, but it's also not very pleasant compound, and we can just generate other peptide bond in peptide. Simple. Then the next step will be coupling of next amino acid. If we have already other group here, we can just put activated amino acid and make a peptide. So, common peptide synthesis protocol. And we made the application starting with some known bioactive peptides, and our strategy needed answer to two questions. The most important question at that time was where to put other group, and then, of course, how to put it, because it was written in textbook that this is very simple reaction. But we were not well educated at that time, and we were looking only some textbooks, but the whole history of other peptide chemistry demonstrates that we have from 1986, there are data from PubMed, until 2025, there are only 241 papers. Very promising topic, but so few papers. The scale is here. Last year it was 13 papers. The maximum is some 40 papers per year. If we take any of these papers, we can see that people have studied influence of other group on some bioactivity property. Let's say stability. There are few papers about cell penetration, penetrant analogs, other penetrant analogs, and some other aspects. But all these papers, some 5-6 papers per year, they are made with very hard work. It was very hard work to get these papers, because in few papers they mention that the yield of other peptide bond was 1%, 2%, 5%. This is nonsense for synthesis of peptide. Some papers do not mention this. So there must be something stupid or crazy from chemical point of view. And of course, we reshaped immediately our grant application, where we planned to make a lot of other peptides, and just made it more realistic that we would like to see why the synthesis is not so easy going. One point why we should, what we should take into consideration is that the other peptide bond has different rotation properties, and therefore the conformation is more flat and conformation is changed, so you can compare the common peptide and other peptide. And the most important conclusion is that it's good to have only one replacement per peptide, or replacement at different sites, not so that we can generate other peptide which has several other groups in sequence. So, okay, that was easy. And now we had a pioneering work, and all these 3 authors are sitting here. Brain Research published other galanin degradation study, where the weak points of this peptide were discovered experimentally, and these weak points are just the best groups or best positions for other substitution. And we synthesized one peptide. We used the shortest version of galanin, which is still active. It's galanin 211. And we knew that it's a bad reagent, bad ligand, but it was short, and it was just more simple to do. And we put other alanine there. And to get this, we needed some long time. I think it was half a year, because we had to collect the product from reaction product, which has only, let's say, 2-3% of needed compound. So, as I told, we changed our application, but we're unhappy, because our referee was probably an educated person, and he has read the same textbook I showed you. Now people ask money to do something that is easy, well-known, and everybody knows how to do this. So our applications were turned down, and therefore we stopped here at this position, making or putting aza-alanin into the galanin fragment. But still, Barbara Koffler and Andreas Lang from Salzburg, Austria, agreed to run the preliminary experiments, and we found the following two things. First of all, this is galanin on R2 galanin 2 receptor, and this is our peptide, azapeptide. Short one, the affinity is 900 nanomole. That's bad affinity. But as the same short peptide has affinity around 150 to 200 nanomole, so we lost only 4 times affinity. Now we are interested how stable is this aza-alanin fragment, but we had no funds. We had enthusiasm, but no funds. And the second more important finding was that if we compare galanin and our aza fragment on receptor 1, then there is no activity. So the subtype activity is there, and affinity is also available if specificity is if the affinity is there if we use a correct receptor. So, quite good things, but now we need to go on with some further ideas. And the ideas are, we have to to make clear what is the reason why we do not get good yields. Because textbook says the synthesis is simple, but we observe the synthesis is not simple. And indeed, if we have a normal peptide synthesis, we can get fast coupling of the next amino acid here with different activating groups. But with other peptide, the reaction is slow, and even during several hours, we get only a few % of yield. This means that there is one point. The reaction between this other group and incoming amino acid is bad. Nucleophilicity of nitrogen must be reduced. And then, of course, it was very easy to look around in organic chemistry. And there is a huge amount of work made by Professor Meyer group in Germany. And this is some kind of summary of this work in Journal Organic Chemistry. And we can see, if we have amine, nitrogen, which is like we have NH2 group in amino acid, then it reacts with extremely strong electrophile. And rate constant is 2.6, 10 to 2. If we put other analog to nitrogens, then it's 0.2. This means that this difference in reactivity is 1,000 times, and this is enough to kill all reactions with azapeptides in synthesis of azapeptide sequence. This 1,000 times difference is in the case of this extremely good electrophile. Amino acid or carbonyl group is not so good electrophile. So the difference should be even bigger. So we have to wonder, why do we have at all some reaction products? We made a summary of our other peptide studies in 2022 and put it so that expectations and reality. On the reality side, we described precursors which are needed for all amino acids we can have, except serine, threonine, and cysteine, where this methodology does not work. And expectations, we just described all the problems which we see with the synthesis or the chemistry step or chemical synthesis of the other peptide sequence. And this was planned as a last slide of my today's talk. But while preparing this talk or polishing the slides, I discovered a paper which is continuation of our work. It's in archives, and they claim that they have made automated other peptide synthesis, case study, and so on. It's American paper. It's made very carefully. It's a chemically very solid paper, and they used some different symptoms instead of this our precursors, but not very much different. But still, they are, of course, they are also using the microwave-assisted analysis, but still they had to use for coupling of other amino acid long reaction times. 2 different couplings, and if the reaction time is 6 hours, 8 hours, 10 hours, this is not the real outcome. But still, they probably can operate their system so that they can do this synthesis. And as you know, in archives, this paper is not refereed. This was sent there at the end of November. So December, January, February, 3 months. So I really hope that the referee is not very educated. It's less educated than our referee was, and this paper will go through. I hope it will be in some good journal. And if I read this paper, they just cite our paper, previous paper, that proceeding from these problems we can now go on with other peptide synthesis in automatic systems. What makes me more confident that this will go through is that they have already some patent application. This means that this must really be interesting for industrial level. So what is the summary? As we have precursors, as we have now technology, I think that maybe now it is much better time to think about other peptide, other scanning, other peptide studies, other peptide synthesis, and of course producing massive papers. My co-workers from Tartu are Anu, Anton, and Meeli, and of course I am very grateful to Barbara and Andreas from Salzburg. So, thank you for your attention.
Thank you, Professor Järv. Do we have questions? Yes.
You are a great man.
Do you have some proof?
May I continue the sentence? If they can make a GLP-1 agonist analog which the original Novo Nordisk patent does not list. And I am sure it's not among the claimed peptides. And they only can make 0.1% of that much as you had as a yield.
No, their yields are quite good.
It doesn't matter what they write. If in reality they can make 0.1%, the product is approved by FDA, and they promise to sell it in Hong Kong for $1.10, and they can sell 6 bags, then Novo Nordisk has to buy that company, even if their yield will be 0.1%. And that's the whole story. You know, I once, in the very beginning at Roche, made a wonderful compound as an anti-epileptic. And we claimed every salt of it.
Yeah, but...
Except one salt, which you can't imagine anybody would ever use. A small company called Sanofi made Roche to pay $100 million. They never made more than 1 batch.
Okay,
So don't be disappointed. It's very interesting. It's just capitalism at its worst, using good science.
But there is one important aspect, Tamas. The FDA, FDA permission is for ligand, but this is a new compound. This has other group in. And this must be validated.
You don't know when they filed the patent, that they published it here. It's not the same date than when they went to the intellectual property office of that Chinese company. They first went there. When it was submitted, they published a paper. The next one will be to make. It doesn't matter if it costs $1 million to make, because they will sell the company for $100 million.
Yeah, but this.
Only 1 month's income of Novo.
But this company is equipment building company. Yeah, not compound. Yeah. Let's see.
But your paper is the key. Somebody showed that this is possible. The yield is uninteresting. If the compound is of value, not dangerous, and has a commercial value, all of this is known already about GLP-1. And from Yakier and his colleagues is known, it's possible to make at a very low yield. And it's no laws against it to be a bendy businessman. So somebody makes it at a very low yield. Until the point somebody is forced to buy that company. And then it's all of a sudden no longer such a bad thing.
But there is one thing missing. Somebody has to prove.
Your paper is key.
No. Somebody has to prove that the other analog has the same activity or is even better.
That's in the patent application. They run the tube and they run the mix. That's in the patent application. That's about 2 months before the paper you read. 60
Let's see.
days at least.
Let's see.
So your paper is great. That's the key. And you will get nothing. Not even a card. Thank you. And if you ask them, why did you send me a card? Thank you. They will say the stamp to Estonia was expensive.
But this is not my patent. So.
This is what happened. If you think of the unthinkable, but it comes out to produce the same biological activity which is approved and valuable.
Thank you for
very funny and very good science.
Thank you for all this discussion. Do we have any other comments or questions? Okay, then I think.
So I think there is a program, and now there was written that concluding remarks.
Concluding
and my
remarks.
name was.
Yes,
So,
yes.
So concluding remarks should be the following. First of all, I would really thank all speakers today who came here to Tallinn and made today's meeting or conference very special. Very special for one reason, because this is the birthday of new pharmacology, as you saw in other peptides. No, it's a new pharmacology, CPPs. Then I thought that I have to make a comment about some secret topics. First idea was that the basic secret is that this conference is connected with Ülo's birthday. But now I discovered that almost everybody knows this. So this comment or concluding remark is not relevant anymore. But there is another aspect. If we, in future, if we ask, when is the birthday of new pharmacology, we can have a very direct connection with Ülo's birthday. So this makes life easier. You can't protest because...
1951, right?
Yeah. Okay, then concluding remarks. I am also very grateful to staff in academy. People were very supportive, helped a lot, and all this successful day, interesting day, productive day was due to their assistance. I'm not mentioning about the beautiful weather, which was specially ordered for today's event. Then, of course, I am very grateful for help from Tallinn Technical University. Because, as you heard from the first speaker, there is quite clear connections. And most importantly, this is already a secret point. We are thinking about writing some future application, maybe EU application or something, research application. And this application needs team. And I really hope that a part of this team is today here in this room. So let's see how things are going, and if the peptide synthesis is growing, then, and including other peptide synthesis, then why not? This application may happen. And finally, I'm really grateful to two societies, Biochemical Society and the Chemical Society, which distributed the information, and that was a great help for organizers to get people here. So, in summary, I think that this was really productive, interesting, and promising event, keeping in mind some future applications and future happenings. So, thank you very much for your patience, for your attendance, and everybody who put energy here in for this input. And as Ülo started today in his report. What is the most important thing? Enthusiasm. And this we saw today in many cases. So, thank you very much, and I think that birthdays must be celebrated, so birthday of new pharmacology must be celebrated in future. Thank you.
I would also ask all the presenters, please, can we make a picture so to have a reminder of this day? So I think in front of the screen there. Õhtul näeme, eks seal Härg restoranis, sinna olete tulemas ju. Jah, kell 6 just.